Honorable dignitaries, learned delegates, respected teachers and students, a very good morning to one and all. I, Ishika Singh,
and Pavitran
feel privileged to welcome you all on behalf of Amity Institute of Microbial Technology with the blessings of our Honorable Founder President, Dr. Ashok K. Chauhan Sir, and dynamic leadership of President and Chancellor, Dr. Atul Chauhan Sir, Additional President and Chancellor, Dr. Asim Chauhan Sir, Amity Global Research Hub, and our dynamic Honorable Vice Chancellor, Professor Dr. Balwinder Shukla Ma'am, to this second day of the two-day international conference on emerging trends in genome editing.
Genome editing fulfills 5 criteria: radical novelty, relatively fast growth, coherence, prominent impact, uncertainty, and ambiguity. Especially when emerging technologies have the potential to change different areas such as agriculture and medicine, the dynamics of the innovation networks from science to business can reach a high level of complexity. The idea of organizing this event is to shed light on the developmental aspects from a technology and innovation perspective.
Well, according to Matt Ridley, life is a slippery thing to define, but it consists of two very different skills: the ability to replicate and the ability to create order. It is on occasion like this we get opportunities to exhibit our talents and to test our knowledge and understanding. We look forward to getting an exposure about what the best of the brains are going to talk about on emerging trends in genome editing.
To begin with our today's session, we have with us Dr. Shashikumar Rode, Senior Scientist and Group Leader, International Center for Genetic Engineering and Biotechnology, New Delhi. He has his PhD in genetics from Delhi University. His area of research includes RNAi, genome editing, CRISPR-Cas9 application in algae, development of algal biofuel technology, synthetic biology, and many more. He was a Ramalinga Swamy Fellow and has received various awards and recognitions from national and international agencies, like Scientist of the Year Award from International Society for Plant Research, Rode Ratna Award for Contribution in Science and Education, to name a few. He is an honorary member of Society for Educational and Scientific Research. He has national and international affiliations from American Society for Plant Biotechnologists and Society for Educational and Scientific Research. He also has various international research collaborations with Ohio State University, University of East Anglia, and University of Pennsylvania. We would now like to welcome Dr. Shashikumar Rode, sir, to deliver a talk on synthetic biology of photosynthetic organisms for industrial applications. Over to you, sir.
Good morning to all, and I'm thankful to Professor Ajit Verma for inviting me for this talk to hear. In synthetic biology, we have focused on two different areas, like carbon capture, which is related to biofuel. In carbon capture, I will try to cover like algae to oil, drop-in jet fuel, bioremediation, biofertilizer, antioxidant. And in plants, we do the drug biosynthesis like terpenoid drugs. So that we can also, and these are the fundings we have for biofuel, like DVT, Reliance Industries, Oil Industry Development Board, Center for High Technologies, Chennai Petrochemical Limited, Tata Steels, and for drugs we have funding from DVT, DST, and National Science Foundation, USA. So first I will like to go to the carbon capture, why it's important. Every year we release around 37 billion ton of CO2 in the environment due to land use, electricity, building, transport, and industries exhaust. So, and who is fixing back this CO2 in the back into the environment? This plant, forest on land, and algae in ocean. These are the one they are fixing back, but they are fixing only up to 50%. Rest 50% is still in the environment. If you take a look at this right side, this world has a finite global carbon budget to keep the global warming under 2 degrees centigrade. If the temperature, if the more CO2 release, temperature will increase, and this even 1 degree centigrade of temperature of Earth will create such a big difficulties on the Earth, uncountable problems will be faced here. So that's why the worldwide there's a total CO2 emission budget around 3 trillion tons. We have used already around 2 trillion tons of CO2 budget. So we left only 1 trillion. And by 2070, we have to we cannot, after 2070, we cannot emit any more CO2. That would be the limit. So our focus is like how we can reduce this CO2 release in the environment by using these biological means, which are the cheapest. So on carbon capture, I have recently given a talk in Canada. So this is on the YouTube. If somebody is interested, can follow this that one. What is the problem with if we working on algae and plant, they cannot fix the CO2 beyond a limit because the Rubisco has efficiency only 25%, and Rubisco has more affinity towards the oxygen rather than the CO2. If there is a less CO2, there is not enough CO2 enrichment into the Rubisco chamber, then it will do the oxygenation rather than carboxylation. If you see the left black diagram here, in carboxylation, CO2 is fixed in the 3 carbon sugar molecule, you can say precursor. While if there is not enough CO2 into the chamber, then there is an oxygenation reaction take place and you lose the 2 molecule of CO2. So this is called photorespiration also. So what we try to do here, we have studied that there is some inorganic carbon transport. So we have engineered LCI-A and LCI-B from different algal species here in marine algae. These 2 inorganic carbon transporters to continuously supply the CO2 to the Rubisco chamber. So this will enrich the CO2 in the Rubisco so that the efficiency of Rubisco can be enhanced and we can fix more CO2. And CO2 is in the environment, but when it comes in touch in the aqueous system in water, it converts to the bicarbonate at pH around. So mostly algae or photosynthetic organism in water, they absorb the bicarbonate, not CO2. So once CO2 is come in contact of water, it will be bicarbonate taken by these green tiny algal cells. And this once it reaches to the Rubisco chamber, again there is a carbonic anhydrase which will convert this bicarbonate back to the CO2, because Rubisco use CO2, not the bicarbonate. So it's quite complicated, but I'm just giving the basic because you are the students here. So this way we have genetically engineered to inorganic carbon transporters in marine algae, and by different molecules like southern blot, RT-PCRs, we have by western blots, we have confirmed the localized the proteins where it is expressing around the pyrenoids, which is a specialized structure found in the algae only. And with the western blot, we have confirmed that this protein is expressing. And moreover, around the pyrenoids, we have observed that there's a thick layer of starch. So it's mean this more CO2 is going inside, so more starch is synthesized, like more sugar is synthesized. Even lipid, you see that lipid has been enhanced double on TLC plate. There's hardly any in wild type cells, any tag is visible, but in transgenic it's visible very well. So that way the productivity of cells has been increased by these expressing these transporters. And then we have, by providing extra CO2 to these cells, we have produced 1 gram per liter per day biomass in algae. And further we have provided this technology. We genetically modify this Tetrus salmis subcordiformis and Nana Chloropis oceanica for this industry, Chennai Petroleum Chemical Limited, to have the higher organism which can produce more biomass. So this is the one part. The second, what we're doing on carbon capture via RNA, we have knocked out some genes which were the blockers in the CO2 capture. So now we had made the algae which can thrive in more than 10% CO2, and this algae die if it's in presence of air. So it needs such a high amount of CO2 that it cannot survive in the air. So that way the GMO concern is contained here, which is not a risk anymore, because if you release this algae in water or in air, it will die. Because this algae need a very high amount of CO2, it is suitable only to thrive into the this exhaust like chimney producing the CO2. So this is suitable only for that. And we have observed that using this approach, the biomass was increased 4 times. And this, we can say this theme is simply, we are working with Tata Steel for this one. This project is supported by Tata Steel because when you manufacture high quality of steels, then there is more production of carbon gases. So, and this there's a 9% of global CO2 is released just by the these steel factories worldwide. And similarly, the cement factory also produce lots of CO2. 6 to 7% of world CO2 is released by the cement factory. So what now cement factory in Heidelberg, Heidelberg cement, which in Sweden, what they're doing, they are recycling this CO2 to the algae, and algae capture this CO2 and produce more biomass, and this biomass is used as a feedstock for the poultry farms or the animals which are the like these milk producing animals. So, and the quality of this end product has been improved because of this algae feeding. So, if like cement factory and this steel factory, this CO2 can be captured by algae, we can reduce at least 16% total world CO2. So, this could be the one initiative. Anybody can participate in this program. There's another program we are working on, this drop-in jet fuel. This is a mission innovation program in collaboration of UK, where we are in a single pot, we have produced alkane hydrocarbons. First, we increase in euglena these fatty acids, and these fatty acids were then converted to alkane by expressing the alkane synthase. So, in a single pot, first time we have shown that the alkane can produce using the euglena. There's like Japanese companies are supported by the airlines are working in this area, and they using genetic engineering, not only genetic engineering of euglena to produce fatty acid. Fatty acid is converted further by transesterification into the biodiesel. So the Japanese people growing this euglena into the Thailand because the temperature and light is better there. Similarly, I believe that India is surrounded three sides by the this coastal land, and very good light and temperature is there. So if we use throughout the coastal area of India, then there is a huge amount of biomass or lipids can be produced for the to fulfill our future biofuel requirement. So this is the one program we are trying to focus. Another program like from Brassica napus, we have expressed this DGAT2 genes into the algal cell to enhance the lipids. If you see that that golden these kind of structures here, the bodies are these Nile strains here showing the more lipid production compared to wild type, and the FIPA was higher. So currently the production of biodiesel from algae cost is double. So what if we can increase the amount of our precursors double? Then maybe we can reduce the cost of biodiesel production. So this is the one concept here, that we can increase the lipid productivity in algae. Sustainability is a big question in algae, that there has been throughout the world there has been a questions that algae based biodiesel biofuel is not sustainable. It's true because there is harvesting, dewatering is the major factor which involved in the algae processing for a biofuel. So what we are proposing here, that we grow the algae in vertical reactors where the light is provided from solar panels, and then this is harvested by this centrifugation you can see, and this centrifugation biomass is cooked like pressure cooker kind of systems, which is called hydrothermal liquefaction, where 1 kg algal celery can produce 460 ml biocrude oil and 137 gram biochar. So biocrude oil can be processed with the existing biorefinery to produce different kind of oil. And the biochar, what we have observed, that if you add with the vermicompost, then tomatoes growth has been enhanced 40 to 50% higher. So that byproduct can be used as a good biofertilizer also. And when you process your genetically modify or synthetic microbes through HTL, then DNA get disintegrated. So there is no GMO concern here. So we have to think about protect the environment also, not only the genome editing or synthetic biology or genetic engineering. So another use of biochar is here is ethanol. To produce 1 liter ethanol, there is a wastage of 15 liter of water, and this 15 liter water we have generated is useless. It cannot be used for any purpose. Bacteria cannot grow in it, and the plants die because the photosynthesis stop. So what we have proposed here, we can use this biochar which we got from as a byproduct while processing this algae, and we can get this byproduct and use to filter this water, and we observe that 96 or 97% water this wastewater can be filtered simply through passing through the biochar containing filter, and then algae and any system can be happily grow in this water. Earlier this water was useless and it has melanite pigment which is a carcinogenic. So and there's one student from Egypt working on this. She just recently submitted a PhD thesis. She wanted to explore the astaxanthin production, so we find from Himalayan foothills that's new algal strains which produce the natural astaxanthin. These natural astaxanthins are better than the synthetic astaxanthins because of bioavailability, and astaxanthin are anti-cancer, and they have, it's a highly like I will say antioxidant activity. I think best in among the other antioxidants. So it has a high value. So this is the first time we have produced this astaxanthin from algae in such a huge amount, which has I think commercial application in future if someone want to carry this work further. Drugs, terpenes. So here we working our focus in drug biosynthesis using synthetic biology in plant is mostly on terpenes. So terpenes are like 5 carbon atoms. If you add these 5 carbon atoms, we produce monoterpenes. It is a cannabinoids. Recently, this is J. Kesling is from UC Berkeley, is a very good friend of mine, and he has been our collaborator in past. So he has completely produced biosynthesis and using yeast. And the cannabinoids that produce, some cannabinoids has the some new properties which are not even produced by plants. So this synthetic biology is opening a new area to produce different kind of analogs in drugs also, if they use diverse host system. So this J. Kesling focus on cannabinoids. Then we are focused on the in C10 further add 5 carbon atoms, we can produce sesquiterpenes, which is a carbon 15, which is artemisinin, very strong anti-malaria drugs. So this I will talk you that we are interested in this one. And if you further add in C15 another 5 carbon atom, you can produce Taxol. Taxol is one of the best drug to produce anti-cancer drug to treat the anti-cancer. I think in market it is maybe sold by different name, but this is also plant products. And in past I came to know that from Himalayan region of Nepal, all these shrubs has been wiped out because to get this Taxol for cancer treatment. So there are hardly any plants available to produce Taxol. So this synthetic biology, metabolic engineering approach can be used here to produce. And recently, using this metabolic engineering approach, J. Kesling produced some compound from yeast, which are used in a beer, and that beer is called Happy Beer, and it's in market now. And I also tried in San Francisco this one. They have established their own factory there. Like if you go to US, there is a pub kind of things that they produce fresh beer. You can eat there and you can have fresh beer. So this kind of concept, I think around Bangalore I have seen, but not in other places. So in isopyrenoid terpenoids, which are the important precursors for different medicines, more than 50,000 biomedical compounds are produced using these terpenoids or isopyrenoid pathway. So here you can see that in plant there's two these IPP producing pathway. IPP mean isopentyl pyrophosphate, which is the precursor of more than 50,000 medicine molecule. So it is like IPP produced by plastid chloroplast also. In cytosol pathway also IPP produced, and they crosstalk also. They can, depending on the need, they can cross from one organ to others. So recently I learned that some people using metabolic engineering of microalgae to produce these isopenoid product cannabinoids. So somebody else work photo capture and put here that algae can also be used to produce cannabinoids. So I'm back to I wanted to say something about the sesquiterpene, which is artemisinin, which is the fastest parasite-killing anti-malaria drugs effective in treating the acute attack of malaria, including multi-drug resistant falciparum malaria. So it is very important, and currently this is produced by Artemisia annua plant, and you might have seen in tobacco plant, in other plant, there is a fibrous kind of structure on the dorsal side, opposite to the sun facing side. Those hair kinds of things in Artemisia annua plant accumulate this artemisinin. So artemisinin production from native plant is very low, but this is important drug because malaria is spread like 80% area of India, like in Africa. This red region is the high risk malaria zone. So we try to produce using this drug into plant by nuclear engineering, and we increase the hair structures, but not enhance much. Then we have recently developed the chloroplast engineering to directly express the genes of artemisinin biosynthesis in chloroplast, because chloroplast has like thousands copy of similar genes and can produce 1,000 times the drugs more than the nuclear genome transformation. So the new platform we have developed. And people has used yeast also in past to produce the artemisinin. But when they convert this, because yeast don't have these 450 important enzymes to produce the artemisinin directly, so they could not express those enzymes, so they produce the artemisinic acid and then chemically convert this artemisinic acid to artemisinin. But in this chemical conversion, they lose around 40 to 60% artemisinin. So Sanofi was trying to produce this drug on commercial scale, never produced a single dose using a semi-synthetic biological approach with the aid of $64 million of Bill and Melinda Gates Foundation. And this manufacturing site is located in Italy, is now sold for some other medicinal purpose. So this was the plant product. So we tried to produce into the plant. So what we have done, we have expressed these 6 genes of mammalian pathway which produce higher IPP through the chloroplast transformation. So, and then another 6 genes we have extended this. Once we have higher IPP precursors, we added another gene.
ये समय कहाँ था तुम? क्लास था क्या?
कल से क्लास था, उसके बाद हेडेक हो रहा था बहुत ज्यादा। कल जब भी हो रहा था ना, मेडिसिन लेने के बाद भी सही नहीं हुई, तो फिर मैं इसलिए नहीं गया।
Oh, okay. Or testing. Okay. I thought something with my voice. So, okay. So total 12 genes we have expressed to produce this artemisinin in double transgenic plants. So we have published this work in 2 journals, Metabolic Engineering and Molecular Plants, and you can see that this one chloroplast pathway to produce IPP is present there. One cytosolic pathway is present there to produce IPP. Then we introduced third yeast malolate pathway into the chloroplast. So now we have 3 pathways. Plant has only 2 pathways to produce IPP. We have introduced a third pathway from yeast to have more IPP, because IPP is the most important part here to produce this secondary metabolites. So, and then we have dragged this higher IPP by introducing 6 another gene into nuclear genome. Like to get maximum morphodyne, we have targeted our gene into the mitochondria with mitochondrial signal peptides. To avoid the glycosylation, we targeted the gene into the chloroplast with the chloroplast transient peptides, and this way we are able to produce the artemisinin biosynthesis. So this work was published into the Molecular Plant, and this drug was further tested by malaria lab, produced in the plants in tobacco, and they observed that the plasmodiums with Sargent ring stage, the RBC infected with the plasmodium with Sargent ring stage reduced to dot kind of structure within 24 to 48 hours. And then on this malaria lab tested our drug on this Babsi mice infected with Plasmodium burgei. So and there they found that this blue line in the bottom, that parasitemia reduced to maximum when these transgenic material given by gavage to these infected mice by gavage has the least parasitemia load. So that way this drug was observed more effective even in the pure artemisinin. Why it was effective better? Because plant has these cell walls, and when you are taking drugs like through Ayurvedic systems, the cell wall protect the degradation of this from the digestive juice of your drug. But when it's going into the intestine and gut, there is a cellulase and there is a bacteria which produce small amount of cellulase. They break down the glycosylated bonds and chew out these cell walls and release this drug directly into the bloodstream. So that way, if drug is produced in plant in a small amount, it is more effective than the pure drug. So this was a concept we given now that instead of producing the pure drugs, we can produce a material which has the like plant material with the drugs and can put into the capsule form. Like in lettuce, what we're doing, we have doing this cycle again, expressing all these in lettuce, and want to put this lettuce in a form of capsule. Like after lipolyzer, you can dry the biomass, and then you can determine the dose and put in the capsule, and then you can store like a normal drugs in form and this powder in a form of capsule. So this is the new concept. We are working on it, and we have expressed in lettuce this drug. So this way it will reduce the cost, and purification cost is the big factor making these drugs costly. So if we can directly produce, use a plant as a platform and capture that grounded biomass, dried biomass in the form of cells and deliver to treat that any disease, then cost can be reduced effectively. So when we publish this work in a molecular plant, it has been like highlighted by different media, and I was interviewed in Rajya Sabha TV. This was first came in the Economist newspaper, London, and then it was 2 months back. It has started spreading in the Indian media also. So suddenly I was amazed that how come this work suddenly becomes so important. But anyway, when we do something without realizing, people may feel it's important. We may taking it like as a normal research part. So can it go? You can directly run that YouTube. Sometimes these newspaper
438,000 deaths and 214 million new cases. The WHO statistics for malaria are horrifying. The staggering numbers are despite the discovery of artemisinin, the most novel and effective therapies against malaria. One of the main reasons of malaria being still a rampant infection is that artemisinin, a drug derived from the leaves of sweet wormwood, is highly limited in its production. While it is very difficult to synthesize this compound chemically, wormwood plants take between 190 and 240 days to mature and yields only meager quantities of artemisinin. But a ray of hope could be the new engineered tobacco plant developed by Dr. Shashi Kumar and team from the International Center for Genetic Engineering and Biotechnology in New Delhi. The team has converted tobacco plants into artemisinin factories which can synthesize the compounds faster and better from wormwood. The researchers have genetically engineered a tobacco plant by inserting 6 of the genes involved in artemisinin synthesis pathway into the cell nucleus of tobacco plant. It was observed that these genetically modified plants yields 0.8 milligrams of artemisinin in each gram of dried leaves after being raised for a mere 60 days. Dr. Kumar has also tested an herbal therapy in which he fed infected mice with leaf tissues from his engineered tobacco plant. It was observed that parasite loads in animals fed the leaves were 2/3 of those in animals dosed with pure artemisinin, indicating that extracting the compound from the leaves may not be really necessary. Researchers aim to extend this study to humans using lettuce plants, as both tobacco and wormwood are toxic. The study, which opens up new avenues of treatment for malaria, has been published in the journal Molecular Plant.
So sometime I realized that these new journalists are better at explaining our work than we scientists cannot do. So how to exit from here? Can I come back to my slides? Yeah, here is, that's fine. So with this, I would like to thank the funding agency, DVT, DST, and this OIDV, NSF, Avan, Infrastructures, CPCL, Reliance, Center for High Technology, Tata Steel for giving the funds to carry this work. And recently we organized this synthetic biology workshop, and there's two participants, one Aarti Mishra, she is a faculty here, and one Khushi Yadav, she is a PhD student in Amethi. So I thought I can decide the connectivity with Amethi also. So with this I would say thank you for the invitation.
Thank you, sir, for your thought-provoking and informative talk. I would now request Dr. Jagriti Tyagi ma'am to felicitate Dr. Shashikumar Rode, sir.
After this? Without question. Yeah. So it's a wonderful talk and wonderful work by you. So when you did this, so this tobacco plant, this is a transgenic plant which you created. Do you want to call it, because you were not calling it transgenic? You did some modifications in this to increase the amount of the attachment. Yeah. So whenever you are isolating the artemisinin, so the other part of the, so you are able to isolate it completely in a pure form.
Partial purification.
Partial purification. So like, because we are trying to fight out, I work on head and neck cancer. So every day there is a fight with the tobacco, stop the tobacco. So how would you think that, because you said that you fed the people also with this particular plant. So how do you think that?
No, we didn't fed people, only up to mice.
So how do you think that you will deal with the tobacco having canny and all these toxicity versus the treatment of advancement?
I understand you are talking about the alkaloids present in the tobacco, whether they are interfering with our drugs. Or
you must be giving it for the very short time. So
I have not. Now I reduce this presentation very small. So we have the control as a pure tobacco also, and that has not control this malaria. So we tested that also that way. And there's a previous report, people tried to express like some other drugs, and they tested that tobacco plants are not involved in controlling malaria, either alkaloids or other compounds present in tobacco. So it's just a model species we use. Now we are working on lettuce. So
Yeah, no. So I'm not saying about the control. I was more worried about the toxicity of the tobacco, or maybe the other effects of the tobacco, which is related to the cancer and all those things. I'm talking about those things because tobacco is a plant. Tobacco causes a lot of say other problems. So that's what I was trying to ask. That do you think that that may also interfere with that, or you're, that's why I asked that if you're isolating artemisinin completely and then you are working, that would be a better thing, or?
No. Complete purification is, we don't want to do that. Because that you can do directly from this Artemisia annua plant also. And the cost is very, very high because of extraction and purification. Very small amount of drug is get purified. That's why this Artemisinin not available to everyone.
Yeah.
So our purpose is to use this model species just to show our proof of concept that we can produce this drug in alternative plant also. So that was the whole purpose. Then we tested that whether this medicine produced in tobacco is functional or not. That we use the malaria lab help.
Dr. Shashi Kumar, Das here. Hello.
Hello.
Excellent talk. Thank you so much. Could you see me? I'm audible?
Yeah, I can see.
Sure. Great to see you here for this talk. Excellent talks.
Yeah.
And so many things you have covered. I have just a minor query. As a cancer biologist, we know the Taxol is long time discovered, and it was derived from the bark of the Pacific yeep tree, available in the Himalayan in India and others. And actually, it is done in US. But you told that it can be the paclitaxel, and Taxol can be derived from terpene. Is that a commercial level? It could be derived from those?
It's I think like it's a under thought process. Like I am also wanted to do it, so I assume that in world there may be 3-4 more scientists may be trying to do the same.
I see.
Try to explore using yeast fermentation to produce Taxol. Now pathways, I think 95% is already known. So once this 100% is known, then many people can produce Taxol in either in alternative plants or the yeast system or bacteria. Bacteria, it may be difficult because then there may be no proper processing and folding of your molecule.
Because of the fact that if it is other way of production is possible, then the price could be much lower.
Yes.
Because it is high cost and not much easily available in India, but it is of course nowadays available because it's a most important drug for many cancer, but mostly breast cancer. So
Professor Silva, where are you hiding? I was hoping to see you here.
Yeah, I came late because of I'm in the office only. I just now reached, but I was on the way and I got the message, and I was listening to you on my mobile. So just you started the talk and I was listening. It was a nice journey and your talk. So I listened almost. Maybe 2-3 minutes I could not. In the beginning, I could not join. So thank you so much. And maybe in lunchtime we'll meet sometime if you are here. But you are in the campus or you are talking from there?
No, I'm in Amity campus.
Okay. Great. I'm in the J3 block here only. Just I reached. So I will come sometime and see you if you are till lunchtime here.
Yeah, I'm here till lunch.
Okay, thank you so much.
Question. So, congratulations, Dr. Rode, for such a nice talk. I have a question that, or rather, a small query, that in few rhizobacterium, there is partial presence of the pathway for artemisinin production. Some of the intermediate steps are, I think, there is some mutation and they are not being synthesized. So can't it be exploited for production of artemisinin, like Azospirillum brasiliense? If I am remembering it correct.
Say again your question.
Means,
That spawn came, so my
so
focus is
basically
changed.
some bacteria like rhizobacteria,
Bacteria
Azospirillum
you cannot
brasiliense.
produce this artemisinin.
If I remember correct, I think Azospirillum brasiliense has few components of this pathway where some intermediates are synthesized and this entire pathway is not present. So if we can complement, can we do that in bacteria?
You saying again that you want to change the mechanism of this bacterial system, because J. Kessling first tried a bacteria. And these bacteria, these terpenoids products, when they produce in bacteria, they kill the bacteria actually. So it's difficult to use. Only eukaryotic system can be explored. Even yeast could not be successful to produce the artemisinin, because the artemisinin itself is a toxic. So what I have done, I produced dihydroartemisinic acid, which whenever you harvest the cells, it come in contact of oxygen and light. It oxidizes to artemisinin. So that's the catch I am using, which I have not told here. Otherwise, it can kill even yeast or bacteria. You cannot produce the
So
pure
whenever
artemisinin.
you lyse it, it comes out and it forms the real artemisinin.
Yeah, so I am using DHA products.
Thank you.
Thank you, sir, for answering all the queries with perfection. Now I request Dr. Jagrati Tyagi ma'am to felicitate Dr. Shashikumar sir. Thank you, Jagrati ma'am. Next, we have with us Dr. Vibha Tandon, Professor, Special Center for Molecular Medicine, JNU. Her expertise lies in translational research. Recently, her laboratory has developed few benzimidazoles as radioprotectors or radiomitigators for radiotherapy of cancer patients and elucidated its mechanism of action in normal cells and tumor cells. Her group has been working extensively on the development of small molecules targeting topoisomerase in bacteria to fight the MDR bacterial strains arising due to mutations in bacterial viruses. She is a recipient of various significant awards and fellowships like Fellow of Royal Society of Chemistry, Professor D. Nasipuri Memorial Award conferred by Indian Chemical Society, Calcutta, DAD Fellow under Indo-German Exchange Program of DST DAD, Royal Society Fellowship at MRC Cambridge, and INSA Fellowship under the Bilateral Exchange Program with Royal Society. She is a member of National Academy of Sciences, India, Allahabad. She has plethora of publications and patents in reputed journals. Now, I invite upon Dr. Vibha Tandon ma'am to deliver her talk. Over to you, ma'am. I request the participants to please wait for a minute until we resolve the technical glitch.
these, all these molecules were showing activity against the lab strains of E. coli also. And if you see, once again, you can see here that the PPEF was showing you the best activity against the E. coli lab strains also. So with all these information, we have taken some of the waterborne E. coli strains. Then we have taken A. baumannii, Klebsiella pneumoniae, Klebsiella S. typhimurium, Pseudomonas, and S. flexneri, Enterobacteriaceae. So we have been, kept on looking at these strains, and in some of the strains the data was very good, and in some of the strains the data was not good, but we did not find any kind of a resistance against the PPEF. So then we have done with the S. aureus, and when we did with the S. aureus, we have found that our data against the methicillin resistant S. aureus and the vancomycin resistant S. aureus was very good. And then we thought that we will work against the S. aureus and we will try to assign PPEF against the S. aureus only. So now the question arises that how does really this molecule work and what it does actually. So if you look at this for the students, that we have a one lane where there is a supercoiled plasmid is there. When you add the topoisomerase, it gets relaxed. So the relaxed DNA goes up. And then when you are adding the ligand only without the topoisomerase, it is not causing any change in the supercoiled DNA. But when you start adding the topoisomerase along with the ligand, that is the PPEF, you can see that slowly this relaxation of the DNA is getting inhibited. So what it suggests that this molecule is inhibiting the bacterial topoisomerase. So then we did the cleavage assay. So what is this cleavage assay? Cleavage assay, I told you that topoisomerase goes and nicks at the one strand of the DNA. So we could see that when we are adding the PPF is increasing the cleaved DNA in the presence of the magnesium, because magnesium is required for the activity of the enzyme. It is a cofactor in this particular, reaction. So then we did the similar experiment with the magnesium in the absence of magnesium, in the presence of magnesium, in order to find out that what is the mechanism of this particular molecule. So as you see here, that there is a mechanism about the magnesium is known, that magnesium actually facilitates to bring the cleaved DNA to the cleaved DNA, which is bound to the topoisomerase, to the another DNA part. And actually these are all movies, but this will not work because the presentation has been transferred to another laptop. So I just wanted to show those movies. But the only thing, the sense which I want to make you here, that the magnesium is responsible to bring the topoisomerase and the DNA together and do the ligation and further process. So what we have found that in the presence of PPF, this process was completely inhibited. So then, as I told you that it was acting on the DNA topoisomerase 1, but it was not acting on the topoisomerase 2 and 4, that is the gyrase. So what we tried to do, that we tried to look into the gyrase assay, and when we did the gyrase assay, you can see over here that gyrase does a reverse job, that it takes the relaxed DNA and it converts it into the supercoiled DNA. So what you can see here, that when this is the relaxed DNA, and when the gyrase was added, it was converted to the supercoiled DNA. But when you are adding the, when you are adding PPEF, it is not inhibiting the gyrase. So it is still the supercoiling is possible. So what I am trying to tell you, that it is selectively acting on one topoisomerase, it is not acting on the other topoisomerase of the in the bacteria. So then we have compared it with the human topoisomerases, and we can see that it was further not acting on the human topoisomerase also. So similarly, we did with human topoisomerase 1, then human topoisomerase 2 alpha, because it was very necessary for us to show that about the selectivity of the molecule, because more or less the topoisomerase have different functions, but their structures are quite similar, say along from the prokaryote to eukaryote. So it was quite necessary it is actually conserved, almost 36% of the structure is conserved among the topoisomerase from between the different organism. So what we did, that we have developed, we have treated the PPF, we have taken the E. coli and we have treated them for a very long time with the PPF, and we have developed a resistant strain on our own, and we have taken the every time we treat with the PPF, whatever is the amount of the bacteria is left, we take them and again we incubate them, and again so like this we have gone up to the 900 generation of the bacteria, and that we started calling the resistant bacteria. So we are calling here K12 sensitive and K12 resistant. So you can see that to these K12 resistant bacteria, what I'm trying to show you here, that the resistant bacteria which was developed because of the PPF treatment is still sensitive to some of the antibiotic, which was a good news to us, that we are not creating some such kind of a strain which is not responding to any other antibiotic. So when you do such antibiotic development, you have to do certain such experiments to prove that how your molecule is actually processing. So then we have taken some of the highly resistant bacteria also. These are all resistant bacteria, and still you can show here that the molecule PPF was showing 16 mg per ml, 8 mg per ml kind of activity there. So when we have taken these resistant bacteria and we tried to see that what are the changes occurred to these bacteria, we could see that the efflux pumps of the bacteria got upregulated. And if the efflux pumps get upregulated, this is not a good news. But in most of the resistant bacteria, the efflux pumps are getting upregulated. So we wanted to see that if we want to increase we want to decrease the MIC and MBC of our molecule, what are the changes are required, and why the molecule is getting efflux. So this was the answer, that because the efflux pump are getting upregulated, that is why the molecule is getting outside of the bacteria. So these are the few, again, the resistant bacteria, and you can see the profile of the upregulated the efflux pumps. So what I'm trying to suggest you here, that efflux pumps are already upregulated in the multidrug resistant strains. So, and you can see here that there are a couple of TOL-C and couple of efflux pumps are getting more upregulated, and couple of them were getting less upregulated in the presence of the PPF treatment. So, and if you compare it with the standard antibiotic like ciprofloxacin, it is more or less comparable to those antibiotics. So now, what we did, that we have taken the efflux pump deleted say bacteria, we have taken the engineered bacteria in which efflux pumps were deleted, and then you can see here that the PPF is getting more, internalized in the efflux pump deleted bacteria as compared to the efflux pump intact bacteria. So this was further we did, then we made the Acinetobacter baumannii topoisomerases. We have taken S. aureus topoisomerase, SA topo 1A, and we have tried to prove that it is working all over the various kind of the strains. It is having the same mechanism, and it is working against the gram-positive and gram-negative both similarly, and its target is same in the Acinetobacter and the S. aureus also. This is again a movie, it won't work. So with all these information, we have started looking into that how actually PPF is not allowing to the molecules to not allowing the DNA damage or DNA breakage, what is happening. So we have done the one MD simulation experiment. This was this is not the simple MD simulation. This was a single molecule MD simulations were done by Professor Naresh Patwari at IIT Mumbai. And if you look into that, it will not show you over here. But what I'm trying to show you here that when the topoisomerase and PPF are bind to each other, then there is a energy difference of minus 18 is increased. Means it is not allowing once the PPF goes and binds to the topoisomerase, it does not allow, there is a gate in the topoisomerase. That gate, that structure is not opening, and that is how the ssDNA, the DNA is not able to bind over there, and that is how the whole process of replication is being stalled due to the PPF. I'll try here if it will work. It will not. So this is a movie, and if you look at that, you can see over here that there is a gate over here. Fine. And this gets opened like, you can say it's like a flower, it's get away there. But when you put the PPF here, this is the PPF which is put over there, then this gate doesn't open. And because it is not opening, it is not allowing the DNA to enter there. So it is not binding there, and that is what 6 kilocalories difference were there. And when even if the E. coli topo and ssDNA bound first, and the PPF enters over there, even at that time also there is a barrier of 18 kilocalories are generated. So why this was very important to show these movies to the students, because this model does not only true for the PPF, we have tried to develop a model which can be utilized for the other antibiotics also, if you are trying to develop the other antibiotics which are targeting the topoisomerases. So if you have a molecule which doesn't allow the gate opening of the topoisomerases, you may not have that can be utilized while you are screening the other antibiotics against the topoisomerases. Then we did the NGS of the genomics and the transcriptomics of the PPF treated molecules, and we have found that some of the genes were getting upregulated, and we have taken the multi-drug resistant bacteria only in this case, and we could see here that the two-component system of the bacteria, carbohydrate metabolism, maltose transporter, and L-threonine degradation. So there were certain genes which were upregulated, and some of them were getting downregulated, and some of them, where you see here the fimbrial protein or the quorum sensing protein or the ABC transporters. So I'm not showing it to you here that this molecule actually doesn't allow the biofilm formation in the bacteria. And why it doesn't allow? Because all these the biofilm making genes are getting downregulated by the treatment of the PPF, and that is why we are not getting that is how the PPF is restraining the bacteria to make a biofilm. So similarly, we did the number of other genes we have found upregulated and downregulated, which have an important part there. And one of them I would like to show here, that PPF actually not allowing the bacteria to utilize the glucose. So it is trying to find out the alternative paths, that how can it utilize get the energy to divide over there. So that was also a good news for us to develop PPF as a drug of interest. So then, as everybody, after doing a lot of, in vitro work, you have to move to the in vivo work. So we did the in vivo say work. And when we did the in vivo work, we have developed two different models. One is called as the systemic infection model. Systemic infection model is a septicemia model, which is a very acute infection model, and where if somebody is having septicemia, the fatality rate is very high. And the another model we developed, which was the neutropenic thigh model. So we developed the neutropenia in the mice, and then we have injected this drug. So in the systemic infection model, you can see over here that when there was no treatment, all the animal died in the 4 days. But when there was a treatment was given, the animals were active, and they just remain they remain alive forever. And when we have taken the neutropenic thigh model, in the thigh we have created the infection, and when we, after sacrificing the animal, we have taken the thigh and collected the colony forming units from the thigh, we could find that there is a large reduction in the number of infection of the molecule as compared to the non-treated one. And as you can see over here, that there is almost, as I told you initially, that this molecule was being subjected by the efflux pump. So we have taken a efflux pump inhibitor over here. And when we have taken the efflux pump inhibitor along with our molecule, we can very clearly see that there is a synergistic effect of these two molecules are occurring together. And we are getting a very high 6 log reduction in the infection as compared to the PPF only. Then we did the same experiment on the SRS also. And when we did the SRS, we have taken levofloxacin as the standard drug, because levofloxacin is one of the drug which is being used for the treatment of the SRS infection. And again, you can see that the PPF treatment has reduced the infection in the thigh model, and it has allowed the animals, almost say 70% of the animals survived up to the 15 days. So then we did the toxicity of this molecule and the pharmacokinetics analysis of this molecule. And we could see that we are trying to fight out with these things, and we are trying to develop the formulation of this molecule because its half-life inside the body is quite low. And then, so on the basis of these things, we can say that we have a molecule with us which has ability to work on the, gram-positive and gram-negative bacteria, which are multi-drug resistant, because it is targeting on a new target, not on those targets which are already resistant, which have been already mutated due to the antibody treatment. And because of this work, we have got the one DBT BIREC project, which is under process, and where CRO, one of the CRO, Anthem Biosciences, is collecting all the GLP, GMP data for us to get the IND enabling data, and after that we will go ahead for the phase 1 clinical trial. This is the place in which JNU where I work. And these are the few collaborators, and I would like to specifically mention Professor Rajni Gan, Professor Madhubala, who have actually provided all the strains, and Dr. Yukching, who have already been, always been encouraging to me to do this work, and DBT and DST for the funding, SCRB for the funding, and now ICMR for the funding. And so this is all about my work. Thank you very much. If there are any questions, I'm open to it.
That was very nice talk. Thank you so much, ma'am. I was just wondering about the upregulation of efflux proteins. So this is a phenomena we see in human cells as well, that the efflux proteins go up, which we connect to stemness. So what's exactly happening in the bacteria?
Nobody have talked bacteria. In bacteria, at least I didn't come across the stemness and efflux. In mammalian cell it is there, but in bacterial cells we have not talked about it. But the only thing I can tell you there that bacteria are very, very smart actually, and they are. The major problem, I do not connect it with the stemness, but the major problem in the bacteria is that every bacteria have some certain different set of efflux pumps. So when you are handling E. coli, there are a different set of efflux pumps. There are certain common one also, and there are certain different one also, and that is why while you are trying to have any therapeutic intervention, you have to take them in the consideration. And as such, if you ask about that the resistance is, I think stemness can be called in terms of the resistance in the bacteria, that the efflux pump upregulation is directly related to the resistance. That indirectly it can be say yes, because I only told you that all multi-drug resistant bacteria have the highly upregulated efflux pumps, but most of the mutations for which the antibiotic resistance is found, that is in the targets. Because you find that whether the beta-lactamase is getting mutated, or the proteins are getting mutated, or topoisomerase, or the cell wall is getting mutated. So mutations are there, which is, so I won't say that the mutations in the efflux pumps are responsible for the resistance. That's what I just want to say.
And you also talked about porins. So what's the role there for porins?
Yeah, so I didn't want to confuse the students, so I didn't tell. So that was a good thing with our molecule, that say porin upregulation, porin is responsible for the uptake of the molecule. So so what bacteria does, that they cause the porin down regulation when any antibiotic treatment is there. So so that is what, again, in the porin there are certain mutations are given, but a direct correlation with the stemness or the resistance has never been given. I have not come across.
Yeah, but even though I find it quite intriguing, because, what kind of, there must be some kind of a sensor or some kind of, transcriptional regulation that is changing either the transcript levels, or, you can think of connecting it maybe with your transcriptomes and proteomes.
Yeah, obviously. So what happened that bacteria, as an SOS response, does everything, not do anything in bits and fancy. They do everything so that they can fight out with any, I will call that, they recognize it as a xenobiotic. So they will do everything, and one of them is the upregulation of the efflux pump.
Kind of an overall defense mechanism.
Yeah. So they always do overlap, plasmid. They do all kind of a thing. They don't do in the say portions anything. So you have to take it in the consideration that whenever you will go to any drug-resistant bacteria, you will find these conditions already there. So in those conditions you have to work, and that is why I showed that data, that even in those conditions we are able to work over there.
Very nice. Thank you so much.
Yeah. Okay. So thank you.
Thank you very much, ma'am, for your enlightening talk. I would now request Dr. Shalini ma'am to felicitate Dr. Vibha ma'am. Thank you, ma'am. I would now request all the esteemed dignitaries, delegates, and all the attendees to proceed for the networking tea break. We'll begin our session after half an hour. Thank you.
Recording stopped.
Welcome back, everyone. We will now proceed towards our next session.
Our next speaker is Dr. The next speaker is Dr. Ankush Gupta, Assistant Professor, Banaras Hindu University. Dr. Ankush Gupta has his PhD from School of Life Sciences, Jawaharlal Nehru University, New Delhi. He has major expertise in developing novel therapeutics and antibacterial agents against Mycobacterium tuberculosis. He has received Smriti Roy Memorial Gold Medal for the Best Teacher of the Institute of Science 2017. He has been awarded with DST Fast Track Young Scientist in the year 2013 from the Department of Science and Technology, Government of India, and DS Kothari Postdoctoral Fellowship 2010 from University Grants Commission, Government of India. He has over 12 years of research and teaching experience. He has carried out multiple government-sponsored research projects, including projects from Department of Science and Technology and Indian Council of Medical Research, to name a few. He has multiple published research papers, review articles, and book chapters in world-renowned publications. We would now like to welcome Dr. Ankush Gupta to deliver his talk on applications of bacteriophage recombinering with electroporated DNA technology in phage therapy. Over to you, sir.
Very good afternoon to all the participants of this international conference on emerging trends in genome editing. At the very outset, I would like to thank the organizing committee members, especially Professor Ajit Kumar Verma sir and my friend Dr. Naveen Joshi for inviting me and giving me an opportunity to present a part of my research. So my today's topic is application of bacteriophage recombinering with electroporated DNA technology, that is BRED technology, in phage therapy. So my talk would be basically divided into three parts, and I have from yesterday I have seen that most of you are students, so I have kept my talk very basic so that you can connect to the basic biology and then find its application. So my talk would be divided into three parts. First is the introduction, where I'll just briefly touch upon tuberculosis and the importance of bacteriophage therapy. Second part is isolation, purification, characterization of novel bacteriophages against Mycobacterium species. And third is bacteriophage recombinering with electroporated DNA, that is BRED technology and its application. Pointer. So coming to the first part, So as you know that tuberculosis is one of the most ancient and deadliest diseases of mankind, and it is one of the leading cause of mortality, especially in lower and middle income countries like India. Some of the famous victims of tuberculosis are represented here. Especially I would like to talk about Ramanujam, who was a genius of mathematics, and he died because of tuberculosis at the young age of 32 years. So tuberculosis spares nobody, and on March 24, 1882, Robert Koch first announced to the Berlin Physiological Society the successful isolation of a causative agent of tuberculosis, which was later named after 1 year as Mycobacterium tuberculosis. So as you all know that almost more than 95% of microorganisms that resides in the biosphere cannot be cultivated in the laboratory. So if you are able to successfully cultivate or culture a microorganism which is a cause of a disease, then you believe that you are able to counter that microorganism, or you are able to tackle that microorganism. So once it was isolated, people were very happy that TB can be very easily treated upon. But still 140 years have passed, and no easy treatment of TB has come, and drug resistance TB has compounded the problem. So TB is basically an airborne infectious disease, and it's world's most famous infectious cause of morbidity and mortality. It is preventable and curable. India is the highest TB burden country in the world, and emergence of drug resistance, which Professor Vibha Tandon has talked about for gram-negative and gram-positive bacterium. I am talking about mycobacterium, and how it is different from gram-negative and gram-positive, I will briefly touch upon. So this drug resistance phenomena along with HIV epidemic has compounded this problem. So basically the life cycle of mycobacterium, here in the left hand side insert you can see that the colony of Mycobacterium tuberculosis, especially which is drug resistance, is highly lipid rich. It's thick waxy layer which is present. And how it is spread? It is spread from a person who is a patient of TB via air droplet infection, goes to a healthy person, especially pulmonary TB, and almost in most of the cases, in 90% of the cases, the immune system, the innate immune defense mechanism takes care of this bacterium and it eradicates it. But there are instances where this bacterium, it makes a kind of symbiotic association with the macrophages. It enters the macrophages and it resides there. These macrophages divide and they form granulomas. These granulomas have lots of macrophages around it, and other components slowly deposit, and it makes fibroids in the lung. It is calcified even. This remains latent for the entire lifetime of the patient. It re-emerges only when the immunity of the person is down. In cases like diseases like old age, disease like HIV, or stress related, or the person is very weak, then immune system gets weak, and this is again activated, and active bacilli come into the bloodstream and they cause clinical manifestations. So the success of this mycobacterium is basically due to its thick cell wall. So as you all know, you have studied that the cell wall of bacteria are basically bacteria divided into three types: gram-negative, gram-positive, and this mycobacterium. So gram-positive has a thick cell wall, has a thick peptidoglycan layer. Gram-negative bacterium have a thin peptidoglycan layer and an outer membrane which is made up of lipopolysaccharide. And mycobacterium cell has a thin peptidoglycan along with arabinogalactin that is covalently linked to the mycolic acid rich layer. So this mycolic acid is fatty acid which is mostly made up of 60 to 90 carbon. And in cases of XDR, it even exceeds 200 carbon chains. So what are the anti-TB drugs? They are two, first line and second line. The first line is isoniazid, rifampicin, pyrazinamide, and ethanbutan. So any patient who has this detection of TB is first treated with this first line drugs. And if the person doesn't respond, then second line drugs are used like streptomycin, cycloserine, ethionamide, amikacin, or ciprofloxacin. So these are some of the targets, like cell wall, cell membrane, Then you have DNA replication, as ma'am was telling about topoisomerases. And then you have the protein synthesis is inhibited. Then you have the transcription inhibitors. And then you have antimetabolites. So these are the different six different classes of antibiotics that are used for the treatment of the bacterial infection. So what is multi-drug resistance? The by definition, patients whose infecting isolates are resistant in vitro to at least both isoniazid and rifampicin. The first line drugs, these two drugs, if a person is resistant to them, he is called as multi-drug. He or she is called as multi-drug resistant TB patient. The extensively drug resistant TB, the patients whose infecting isolates are resistant in vitro to isoniazid and rifampicin plus any fluoroquinolone and at least one of the three injectable second line drugs like amikacin, kanamycin or capreomycin. So you have resistance against one means first line as well as the second line drugs. So as per the global tuberculosis report from WHO, that is the just before this COVID pandemic 2020, they estimated that 10.4 million people are infected by TB every year. Mortality is 1.5 million deaths globally, with at least 3 lakh people which are compounded with HIV infection also. In 2020, 30 high TB burden countries have accounted for 86% of the TB cases, and 8 countries account for 2/3, with India leading the count, followed by China and Indonesia. So in India, in 2020, India accounted for 26% of the incident TB cases across the globe. So after this coronavirus pandemic, this notification, and it is believed that the notification of TB as well as its drug resistance phenomena will steeply increase. And the latest report of WHO indicates that this has increased. So drug resistance in Indian scenario, 3% of new TB cases are MDR, 12 to 30% cases are retreatment cases, and more than 50% of the global burden is shared with China and the Russian Federation. XDR TB is also on the rise, and approximately 10% of the people with MDR TB have XDR TB also. So you can see this map, all the orange colored regions, they are having high incidences of tuberculosis and drug resistance tuberculosis. India is leading the count. You can see here, India is leading the count. So this problem is our problem and we need to tackle it, because in Western countries you don't have those much infectious disease because of the weather and rich food diet habits and strong immunity. So this problem of drug resistance in tuberculosis is mostly of Asia and Southeast Asian countries. So drug resistant TB is transmitted in the same way as drug susceptible TB. It is divided into 2 parts. Primary resistance refers to initially infected with resistant organisms. Those patients which are first of all infected with a resistant isolate. And acquired resistance that develops during TB therapy. So who are the person who are at increased risk of drug resistance? The person who have a history of treatment with TB drugs, a person who has a contact of person with drug resistant TB, or the person who has smears or cultures which are positive despite 2 months of TB treatment, or the person who received inadequate treatment regimens for more than 2 weeks. So what is this inadequate treatment? So this is basically multifactorial and it is a burning problem in Indian scenario. So we often see that we take, doctors prescribe the antibiotics for say 5 days, 7 days, and what we do, we just take it for 2 days, 3 days. When we feel better, we leave it. And that is the major cause of drug resistance. So what happens? There is irregular or incomplete course of treatment. There is prescription of wrong dosage or length of time. So we have some quack doctors in rural settings who do not know the entire thing and they prescribe wrong duration of drugs, mostly in the rural settings. We have what we call as jhola chap doctors in rural settings. What they do? They prescribe drugs. Indiscriminate use of antibiotics. Then there is insufficient supply or availability of poor quality drugs. Sometimes expired drugs or substandard drugs are supplied in rural settings. Then you have drug misuse or mismanagement. What is there in India? Availability of over-the-counter Schedule H drug without prescription. You go to the counter, any chemist shop, you ask for antibiotic and they will give you. But this doesn't happen in Western countries. You need a prescription to get an antibiotic. So what happens with this? When you have inadequate treatment, you have mutations. This mutations, you have persisters. As ma'am was telling, Vibatandan ma'am, you have persisters. These persisters have mutations against this antibiotic in the DNA. This is transferred to RNA and then protein, and then defective proteins, which are the 6 major targets which I told about the drug. They are mutated, and finally the microorganism becomes drug resistant. So what are the steps which Indian government has taken? Basically, it was declared as global health emergency in 1993. In 1997, RNTCP was launched with DOTS program, where a qualified medical practitioner or trained medical practitioner would observe the person for the treatment duration. Then PMDT, and again in 2012, a major step was taken that it was made notifiable disease. Any doctor whom a patient approaches and is diagnosed with TB has to be notified to the central repository, and this central repository is represented in the global tuberculosis report of WHO. So this and in 2017, this government developed a national strategic plan for TB elimination with sustainable development goals to eliminate TB by 2025. So government is very proactive and it is doing its all to remove this pathogen, but it's very difficult because very few chemotherapeutic compounds are available for TB treatment, and they are administered in combinations for extended periods. So for all other infections, you have a treatment duration of say 1 week, 2 weeks, maximum 15 days, or at the most to a month. But for TB, this treatment goes on for 9 to 12 months. And you can't miss a single dosage. And the one major drawback is that no detailed studies are undertaken in India to investigate the molecular mechanisms for drug resistance in TB in the Indian scenario. What are the major drug resistance pattern which is coming in India? So after the discovery of penicillin, starting from 1930s, there was a boom for this antibiotic discovery from 1940s to 1970s. Major antibiotics were discovered. In 1990s to 2000, few more antibiotics were discovered. But what's now? No new drugs are coming in the pipeline. And it's very difficult, as Professor Vibha Tandon ma'am told, it's very difficult to design a pipeline and to go from discovery up to the treatment stage of any antibiotic. So bad bugs, new drugs. So are we reaching a stage where we are going to encounter a superbug, a strain of bacteria that is resistant to all antibiotics? Is it so? So if it is the situation, it is very deadly. So what are the alternatives? One of the alternatives to antibiotics is bacteriocins. So bacteriocins are basically proteinaceous compounds which are lethal to bacteria other than the producing bacteria. So one strain produces it, the other strain is affected by it, not the producing strain. So usually bacteriocins form pores in the target cells and the target cells are removed, eliminated. Next is antimicrobial peptides. In general, antimicrobial peptides are small molecules with a molecular mass of 1 to 5 kDa. Their structure usually contains elements that facilitate the interaction with negatively charged membranes, and their mode of action involves cell membrane of the target organism. So these also form pores and they kill the target bacterium. Then you know that probiotics are there. So probiotic doesn't kill a bacteria, but they are friendly bacteria and they benefit the host. How? Just a few of the benefits of probiotic provides are by promoting absorption and production of vitamins and enabling efficient inflammatory responses. So probiotics are basically helpful in improving the immunity of a person. So now silver nanoparticles. The nanoparticles have emerged as antimicrobial agent against multiple drug resistant bacteria because of their high surface area to volume ratio and unique chemical and physical properties. However, they have non-specific bacterial toxicity mechanisms. So they sometimes compound the problem. So they are not effective in all the cases. So this is also has limited applications. So what is most promising today at this point of time? The most promising ones are bacteriophages. Bacteriophages are basically obligate intracellular parasites that multiply inside bacterial hosts by making use of some or all of the host biosynthetic machinery. They are viruses that infect bacteria. You might have studied about bacteriophages in your classes, and they are very useful and even therapeutic also. So what is basically a bacteriophage? It is a type of virus that specifically infects its bacterial host. They are also called as bacteria eaters. And they do so by injecting their genetic material, which is mostly double-stranded DNA. So these are one of the most common and diverse entities in the biosphere. So phages are everywhere. The world is a phage and we live in the sea of phages. So phages are the most abundant entities. Approximately 10 to the power 31 different phages are approximated to live in the biosphere. So phages are widely distributed in locations populated by bacterial hosts. River Ganga is a rich source of bacteriophage. You might know from your parents and grandparents that once you keep Ganga water, Ganges water, for months it will not be spoiled. It is very active, and because of bacteriophages. They are the source of bacteriophages. So they are also present in hospital sewage, garden soils, etc. So it has been estimated that there are more than 100 different phage species and at least 10 phages for each bacterium. So based on the morphology, there are 3 types of bacteriophages in the order Cordovirales. Cordo means tailed viruses. These are Myoviridae, which have long contractile tails. Syphoviridae, which has long non-contractile tails, and Podoviridae, which has short stubbed tails. So this is very basic lytic and lysogenic cycle. You might be knowing it. Lytic cycle is when the phage enters the host bacterium and it synthesizes the components of its structure and assembles them and lyses the cell and released. In lysogenic, it basically introduces its DNA, which lives as a prophage by integrating into the host genome and replicates for several generations. So there are some limitations of the phage therapy also, that you cannot use phage therapy indiscriminately. You need to know the biology of the therapeutic phages. What do you mean by biology? You have to know the genomic DNA sequence and the structure of the phages. So what is the genome complement of that phage? You have to know and you have to characterize it. So the phage preparations that you are using should contain only infective phages, and it should be free of bacterial remains. So whatever you are preparing, it should be free of bacterial host remains. And phage receptors on the bacterial surface should be known. The efficacy of the phage therapy should be tested on animal models. So before jumping onto humans, you have to go through an animal model system. Then reversion to lysogeny, leading to DNA integration, should be avoided. So what happens is, if it is a lysogenic phage, it will go and integrate and it will not kill your bacterial host. So lysogenic genes should be removed. So there are some ethical issues which against the use of this live phage particles in human subjects that has to be taken care of before we can use. So how do bacteriophages kill their host? They have a cassette of genes which are called as holin endolysins. Holin forms a multimeric channel in the inner cell membrane through which endolysins pass, and this endolysins renders the wall of the bacteria very weak, and they burst the cells through which the cells die and the active viral particles come out. So endolysins these are phage encoded muralytic enzymes produced by bacteriophages to cleave the host cell wall during the final stages of lytic cycle. So mycobacteriophages have 2 classes of this endolysins. These are one which are against the peptidoglycan layer and other against the mycolic acid rich outer membrane that destroys the arabinogalactin peptidoglycan layer. So these 2 classes of lysins are lysine A against peptidoglycan layer, lysine B against the mycolic acid rich outer membrane. So these endolysins in their pure form are also used as enzybiotics. As in place of antibiotics, they can be used as antibiotics. So they are named as enzybiotics. So they have novel mode of action. They kill antibiotic resistant bacteria also, and they have low probability of developing bacterial resistance. So here you can see that lysine A attacks on peptidoglycan layer, lysine B attacks on the mycolic acid arabinogalactan layer. So the summary of this part is that bacteriophages are harmless to humans, animals and plants, and specifically kill their bacterial host. After their discovery in 1930, phage therapy was abundant. But when antibiotic resistant bacteria, they came into the picture, the therapeutic potential of phages have renewed attention. They co-evolve with their host, and there is least chance of host resistance development. It is essential to characterize the phages before subjecting them to therapy. So challenges are ethical issues, lytic lysogeny conversion, host-free phage samples, etc. After addressing these issues, phage therapy can be put to use. So with this view, we try to isolate bacteriophages against mycobacterium in our laboratory so that we can prepare a library of this mycobacteriophages, that is phages against mycobacterium. So coming down to the second objective. So how phage isolation is done? So you have to collect the sample, which is mostly a moist soil from different environmental niches. Then you have to mix them with phage buffer and filter it with 0.22 micron filter paper. This filtered sample is mixed with host and plated onto the double layer agar plates. So this is very interesting work because whatever you get, because phages are the entities which have high genetic diversity and novelty. So characterization of Kashi VT1. So the phages that we are isolating, that is from Varanasi area. So we are naming them as Kashi series, and whatever the place that we are isolating them. So here you can see that you have the plaques which are developed on the plates. So these are against the Mycobacterium smegmatis host. Smegmatis is a non-pathogenic host of Mycobacterium. Mycobacterium tuberculosis is pathogenic, but for developing those phages against Mycobacterium tuberculosis, you need to have BSL-3 facility. Okay, biosafety level 3 facility. But in normal laboratories like BSL-2, we have BSL-2 facility, so we isolate against Mycobacterium smegmatis. So this is isolated from Vishnath temple, famous Vishnath temple in BHU campus. And this belongs to Cyphoviridae family. So we did a genomic sequencing of this phage, and we found that it has a 61 kb genome, and it belongs to cluster K1 with 66.5% GC content. It has 101 ORFs, and 41 gene products are functionally predicted, whereas 59 were assigned as hypothetical protein. So this is the point where I want to say that these phages are very rich genetically. There are several genes which need to be characterized. So it has one tRNA, and this we have submitted to the NCBI database with the following accession number. So this is the structure, TEM structure of this phage. You can see that it is Cyphoviridae basically, and it has approximately 275 nanometer in length with a head of 70 nanometer approximately and tail 200 nanometer. So on the restriction digestion analysis, we found that it has a rich PST1 sites in its genome and 1 or 2 BAMH1 sites. So then we characterized it with respect to the multiplicity of infection. What is multiplicity of infection? It is the ratio of number of virus particles to the number of host particles in a defined space. So that is PFU per ml upon CFU per ml. So here what we do is we keep the phage concentration fixed, and we decrease the bacterial host concentration 10-folds to see which is the best concentration of the phage to virus ratio, that is phage to bacterial host ratio, which induces its synthesis. So we found that 1 phage per every 10 bacterium is very good for this bacterium. So we did the stability studies for temperature and pH. We incubated this phage for 1 hour at different temperatures and found that it is stable up to 55 degrees centigrade, and it is stable at a pH of around 8 and 9. It has the maximum activity. So One-step growth curve assay was also done to see the burst size and latency period. What is the burst size? The burst size is basically the average number of phage released per bacterium is called the burst size. So the burst size for KVT1 was 102, and latency period, that is the time between the injection of the viral genome into the host and host cell lysis, was 90 minutes. So this phage was active after 90 minutes, and then it was giving a huge round, approximately 100 phages per one bacterium. So this is the fluorescence microscopy data. Here you can see these blobs coming, these blue blobs. So we stained it with Nile red and DAPI. DAPI stains the nucleus, nucleoid region basically. And whenever this blobbing occurs, this means that the cell is going to die. And here you can see that maximum blobbing occurs at 60 minutes. So here the cells are dying, and you can see the cell debris at 120 minutes of time. So we also isolated another phage which we named as Kashi SSH1. This phage was isolated from Sir Sundarlal Hospital, which is present in the BHU campus, and it is from myoviridae. You can see the plaques of this virus. So upon the DNA sequencing, we found that it is surprisingly, it is very huge. It is 155 kb approximately. These phages are generally 50 to 60 kb, but this phage was 155 kb, and it has 237 ORFs. 49 gene products are functionally predicted, and 188 are assigned as hypothetical proteins. So you can see that approximately 200 genes do not have known functions. So it is novel, and there is an opportunity to characterize them. So 37 tRNAs and 1 tmRNA has been identified, and it has been submitted to NCBI database with the accession number. So genomic restriction digestion also, this virus also had a lot of PST1 sites and 1 or 2 BAMH1 sites. So here you can see the structure. It has icosahedral head and tail which is shorter than Cyphoviridae, and it is basically myoviridae, and this is short, long, non-contractile tails basically. So it has even better MOI. It works at 1 phage per 100 bacterium, so it is more infective phage. So it is also stable up to 55 degrees centigrade, and maximum active at pH 8 to 9. So its burst size is even greater and double than KVT1. It's around 200 phages, and its latency period is 60 minutes. So here also you can see that the blobbing starts around 60 minutes, and around 90 minutes the complete bacterial host is eliminated. So bacteriophages have been, KVT1 and KSS1 have been isolated against non-pathogenic bacterium host, M. smegmatis. KVT1 and KSS1 have been characterized with respect to several parameters, and they have to be tested against other pathogenic mycobacterium including MTB. So if non-infective to MTB, genetic mutations can be applied to introduce infectivity in these phages against pathogenic mycobacterium species including MTB. So this have not been tested against MTB as a pathogen, but once we test it, we can use it against Mycobacterium tuberculosis. So now coming down to the bread technology and its application. So this bread technology basically works on recombination. So quickly to revise your classroom data. So genetic recombination is the exchange of genetic material between different organisms of a species, which led to production of offsprings with combination of traits that differ from those found in either parents. So in diploid eukaryotic organism, there is an exchange of genetic information between newly duplicated chromosomes during the process of meiosis. Recombination also occurs in prokaryotic cells, especially well characterized in E. coli. E. coli doesn't undergo meiosis, but they have a type of sexual reproduction called conjugation, where the genetic material is transferred from one bacterium to another and may be recombined in the recipient cell. So there are 3 types of homologous recombination basically. Exchange between similar DNA molecule, Then there is illegitimate or non-homologous recombination, and then site-specific recombination. So homologous recombination is very important because the genetic information is exchanged between similar or identical molecules of double stranded or single stranded DNA. So what are its applications? Its application is in DNA repair, DNA replication, fork issue, and meiotic chromosome segregation and telomere maintenance. So this is basically the mechanism of homologous recombination that I'm not going into the detail. So in bacteria, this is important. So in bacteria, how does recombination occurs? There is a specific pathway in your E. coli called REC-BCD pathway and REC-F pathway. There are Chi sites which are recombination hotspots that occur in every 1 per 5 kb DNA. So what happens, this REC-BCD is an endonuclease, exonuclease. It degrades one of the strands, and then REC-A works as the homologous, works in the homologous recombination. And RUV-ABC, they are basically, they resolve the holiday junctions. How does recombination occur in phages? This is important. Now here we are coming into the phages, and this phage technology will be adapted into bacteria for recombinating. So the most common and well studied is the lambda red system in bacteriophage. So it has basically 3 proteins, exo, beta and gam. Exo is a 5' to 3' nucleases. It binds to a double-stranded DNA, cleaves and makes a single-stranded 3' overhang upon which beta binds and does the homologous recombination. So this gam basically inhibits the bacterial intrinsic REC-BCD pathways. So why bacterial recombination is not used for recombinating? Because it is less efficient. And phage proteins used for recombinating because they enhance the frequency of homologous recombination. So now coming down to recombinating. What is recombinating? It is the engineering of recombinant DNA by homologous recombination. Recombination systems encoded by bacteriophage, they enhance the frequency of homologous recombination. It was originally designed for modification of E. coli. Shuttle phasmids have been used, but they involve complex genetic constructions and multiple steps of manipulation and screening. So here you can see how shuttle phasmids work, but they take a lot of time and they are cumbersome in preparation. So RAC prophage. So this is a prophage which is present in E. coli genome. It also does homologous recombination. So it has 2 proteins, REC-E and REC-T. So in vitro, the DNA substrate is prepared and it is transformed into bacteria where REC-E binds as an exonuclease and REC-T does the homologous recombination. So what are these applications of recombinating? That you can change the bacterial host as well as the phage by point mutation, in-frame internal deletion, insertion, replacement and gene knockout. So the need comes for mycobacteriophage mutants by recombinating. So why to prepare mycobacteriophage mutants? It is because we want to alter the host range. That is, we want these mycobacteriophages to infect from non-infective bacterial host to infective bacterial host. That is pathogenic bacterial host. So what we are doing is we are isolating against the non-pathogenic bacterial host. We want them to bind and kill the pathogenic bacterial host. So that's why we have to alter the host range by introducing genetic mutations in this phages. So removal of lysogenic determinants and preparing reporter phages for detection and diagnosis of the disease. So shuttle phasmids cannot be used in mycobacteriophages because their genomes are too large. Few mycobacteriophages form stable lysogens and relatively poor DNA uptake by their hosts. So bread technology is developed. In the bread technology, this genome of the bacteriophage is isolated and double-stranded recombinating DNA substrate, which is linear double-stranded substrate, they are co-electroporated in a bacterial host where you have already expressed REC-E and REC-T homologs. REC-E and REC-T are basically involved in homologous recombination. So you have recombinant phages which are being released by the host bacterium. So in mycobacteriophage, the homologs of RAC prophage system, that is REC-E and REC-T, are very rare. We can say that these identifiable recombination proteins are very rare in mycobacteriophages. it is only 3% of the mycobacteriophages have this recombination systems till date. And maximum work on this has been done by Professor Graham Hatfull from Pittsburgh University. And he found that for every 30 phage, he gets one mycobacteriophage which has this recombination system. And that too, that are not incomplete. Either Rec-E homolog is present or Rec-T homolog is present. So he basically found a bacteriophage called Che9C, and this bacteriophage has been found from India, from Chennai, by Professor Vanaj Kumar, and he has donated it to Graham Hatfull. Currently, Graham Hatfull has around 2,100 sequenced mycobacteriophages, but very few have these. So while working on these mycobacteriophages, we found that we are getting much higher percentage of bacteriophages which are having this recombination machinery. We are having 20% efficiency. We have isolated approximately 10 mycobacteriophages, and among them we are getting, in 2 of them, we are getting this recombination machinery which is full. So we are doing the next work on recombinating with those bacteriophages. So this is basically how you will screen the mutants. You will screen it with PCR. So screening is also not very difficult. So this is one of the latest. It is in very infancy. This work has been done in 2019, where they have treated Mycobacterium abscessus, this Graham Hatfull's group, and they have used engineered mycobacteriophages. You can see that these skin lesions are formed due to Mycobacterium abscessus. 3 phage cocktail has been used, and their lytic phages derived by bread technology has been used. And intravenously, this phage has been injected, and substantial resolution of infected skin nodules are seen. That is, it is mostly removed. So these are the 3 phages which are of Cyphobacteridae family, and this cocktail has been used against this bacterium, and it has been successfully employed. So we have a recent publication this year. So this is the second report against Mycobacterium chelonis. Here they have used a single phage therapy. This is also a cutaneous infection. So it is highly disseminated. A single phage therapy is used in IV mode, and but it developed some neutralizing antibodies. But there is substantial improvement of disease with negative biopsies and no evidence of bacterial resistance to the phage. So here you can see that it has been improved very nicely, and this phage therapy is working. So this bread technology along with bacteriophage therapy is in its very infancy. It has been used against other species of Mycobacterium, but Mycobacterium tuberculosis is still a challenge, and we need to develop. The first challenge is, can we isolate more novel mycobacteriophages and then introduce mutations in them so that this can be used for bacteriophage therapy against tuberculosis. So basically, bacteriophage bread technology represents a very promising approach for directed evolution of bacteriophage genomes through targeted mutations, that is to alter the host range, removal of lysogenic determinants, construction of reporter phages, and this can be used for phage therapy either singly or in cocktails. Thank you so much. So this work, I would like to thank my students. This is my group which is working there. So these girls are basically in the phage biology group, and these boys are in the proteomics group. So this girl, Tanmay, has initiated this work in my lab. And after 3 years of intense hard work, she was able to isolate these mycobacteriophages, because the protocol is not that easy. You can get phages against gram-negative and gram-positive very easily, but getting phages against mycobacterium is very difficult. So she tried a lot of combinations and buffers, and then she developed. The recombinating work is done by this work, and this girl and this girl. So this is my collaborator. Whatever I was telling about Graham Hatfull, he has donated me some phages, 10 to 11, to work upon with genetic engineering. And this is Dr. Louise Temple from James Madison University. She has helped us in getting those genomes. We were getting raw data from the company, but we were not able to assemble and annotate those genomes. So she helped and trained my students in annotation and assembly. So this funding is from ICMR and UGC IOE, and JRF and SRF to the students from ICMR, UGC, DBSP is highly acknowledged. Thanks. So this talk was very basic, and if you have questions, I would be happy to answer that.
I think that's a very daunting work. I mean, you're trying to change the way people look at treatments, because phage treatment it's definitely issues with it, like if just a conversion that you talk about, right? So firstly, what I did not understand that like CRISPR-Cas was being found in bacteria, right? And now we are talking about virus, where we can see a similar kind of mechanism of recombinating. But is it also a defense mechanism for virus? Because normally what we learn from CRISPR, we see that it's a defense mechanism for bacteria.
It's basically a defense mechanism for bacteria.
Yes.
But in case of E. coli, definitely there is this defense mechanism, but not much information about the CRISPR-Cas9 mechanism in Mycobacterium is there. So it is not very highly functional. Even this, the REC-E and T homologs that I am telling is very rare. It's very rare. But fortunately, we have got some phages, and in India, the phages that we are getting, they are highly diverse. You have that genetic complement which can counter the bacteria.
Yeah, so I'm also not able to understand that, maybe it's new for me, that recombinating, you are trying, that those parts are available in the phage. Or it's there in the phage, maybe full or not full.
Yeah.
And then, because virus does not work by itself.
Yes, it will not.
It need a host.
Yeah.
So it goes into the host and then recombination happen. So can you just explain the mechanism there?
Yeah. I skipped it quickly. So here you can see, or even better here. It will be this one. So what happens is, these are the host bacterial cells. Once we transform these cells with plasmids, which are having this bacterial component of REC-E or REC-T homolog, they are a cassette. This cassette is cloned in an inducible promoter, say acidamidase promoter, and then this is transformed into the bacterial host. Then this bacterial host is induced with the inducer, and then you take out the phage DNA, which is isolated somewhere else. This phage DNA along with a linear DNA substrate, which you want to incorporate into the phage, or if you want to mutate even the bacterial host, you can mutate with this technology. So you have the enzymes which are coming from the phage. These are the phage enzymes which are already expressed. Once you electroporate, you get plaques here. You take individual plaque, you screen it with the help of these flanking primers. So either you get mixed plaques. So you get wild type also, you get mutant also. So what you do is you go for second round of screening, where you dilute it serially, tenfold up to 10 to the power -6 concentration, and you get mutant pure phages from that. And this pure mutant phages can be used for phage therapy or whatever your objective is. You can make fluorescent phages, you can change its host range, or you can delete the lysogeny related genes. So these are what are required, and once you mutate them, you can use them as cocktails, like Professor Graham Hatfull has used. And this phage therapy is not normally recommended. It is only recommended when you have at least 10 to 20 antibiotics which are resistant. So these two cases which he has reported, at least 20 antibiotics were used for that, and the patient was not responding, and heavy toxicity was there in the patient, so they were recommended by the ethical committee for phage therapy, and then the patient recovered.
And any follow-up study on the patient, like any kind of side effects or toxicity or anything?
No, there was some sweating was there. This they have mentioned in the paper, since this is a very recent paper, 2022 and 2019. 2019 patient is okay. 2022, no follow-up, because this is a recent paper. So this is in very infancy, but this is very promising. So we are trying for that, because once you get it against tuberculosis, it is a very big deal for India.
Absolutely. Thank you so much.
Thank you.
Ankush? Yeah. So, nice talk, Ankush. It's really great work you're doing. My question is a bit basic, actually. I'm not a virologist, so I work in plants. So you isolated these phases from some places around BHU. So how you, because basic virology, we know that all the viruses, they are the host specific. So how you determine that the phases you isolated, they are specific to mycobacterium?
So we use a screening host basically. What we do is, since we have a BSL-2 lab, so what we do the screening with Mycobacterium smegmatis. It is a surrogate host which is non-pathogenic. So we have pure cultures of Mycobacterium smegmatis. What we do, we take out the environmental samples, we mixed it with a special composition of phage buffer, and we get it filtered. First we centrifuge it, and then we filter it with 0.22 micron filter paper, and then this filtered substance is then spotted along, it is mixed with the bacterial cells, and then it is plated on a double layer plate which has upper layer, agar layer, and a lower layer. So upper layer has low agar percentage, so there they form plaques, and these plaques are, individual plaques are lifted, and then these are amplified. So purification happens, and we are trying for getting it against tuberculosis. So BSL-3 also, I am trying to put one student on BSL-3 facility for sub-screening of this, because few phages are monovalent, which works for one strain of bacteria, and few phages are polyvalent, which works on, say, 2 or 3 different species. So we are trying for that polyvalent phages, and all these phages have to be tested on MTB now.
Thank you. Thank you very much.
Thank you.
Thank you very much, sir, for sharing your deep knowledge with us and for answering all the queries very precisely. Now I request Dr. Manpreet ma'am to please felicitate Dr. Ankush sir. Thank you, ma'am.
For our next speaker, we have with us Dr. Manoj Kumar, Associate Professor, Amity Institute of Genome Engineering, Amity University, Uttar Pradesh. Dr. Manoj Kumar completed his PhD from Max Planck Institute for Plant Breeding Research and University of Cologne, Germany. He pursued his postdoctoral research at John Innes Center, Norwich and Sainsbury Laboratory, University of Cambridge, and studied how plants sense and respond to temperature. He contributed to the discovery of how phytochrome B senses temperature and regulates plant growth. He further demonstrated chloroplast signaling gates thermotolerance in Arabidopsis. His current research is focused on understanding the gene regulations in plants and develop plant varieties with better nutritional resources. He has published plethora of research papers in renowned publications. Now, I invite upon Dr. Manoj Kumar to deliver his talk on identification of dynamic sensors in plants and their transcriptional regulation. Over to you, sir.
very good afternoon to all. And I understand that this is going to be lunchtime. The students must be feeling hungry, and I can tell you that I have just 20 slides. Only 20 slides, so please bear with me, and it will be fun. So, with basics and a bit of details, both will be there. So, first and foremost, I would like to thank the organizers, our respected Professor. I am sorry, so I am struggling with this. Professor Ajit Verma, sir, whom I know for 20 years. I am sure he must remember. It was my first book chapter which I contributed in the book edited by him. And Dr. Naveen, who invited me as well in this, and we were doing our PhD studies and other studies in JNU together with Dr. Ankush as well. So thank you so much. And before getting too late, I should start. So before we go in much detail, I belong to a very newly established institute, MET Institute of Genome Engineering. It's almost just 1 year, few months, which has been founded on 14th of May with the kind blessings of our honorable founder president, sir. He has said something, and you can have a look on this website, met.edu/AIGE. So it is headed by, at the moment, a very able mentor, Professor V.C. Das, sir, and he has a number of creative ideas to push the new domains of this technology. And I have few colleagues over there, Dr. Ramesh Thimappa. He is a Ramalinga Swami Fellow, and another one is Dr. Mohammed Jamseer K. Some of you might have seen them walking around in J3 third floor. Jamseer at the moment is in Lausanne, Switzerland, pursuing his postdoctoral, and he is on leave for time being. So during last 1 year, what have we done? So we have ongoing grants, 7. We have only 11 publications, although the cumulative impact factor is 93. So out of 10, they are in Q1 journals. Some of them I have put it. One is in EMBO reports, B1 type cyclins control microtubule organization, trends in plant science, Journal of Experimental Botany. We also published our first genome-edited plants-based report in Plants Journal targeting TOR and SNRK1 gene in rice with CRISPR-Cas9. The work was done by Jamseer. And very recently we have published in Nature Chemical Biology. First author and lead author is Dr. Ramesh Thimappa. So you can see that we are really doing a class research. And I can say we are doing at par with any other institute in India, if not abroad. So at the same time, this year on, we have vacancies for PhD students, and we have also started Master of Science in Synthetic Biology and Genome Engineering, and I'm sure some of you would like to join there. Seats are limited though, only 10, but I would welcome any capable student who would like to pursue master program in this cutting edge, really cutting edge, and we have all facilities. That's why our seats are only limited, 10, so that we can train all students. So coming back to science of mind, for which I am here mostly. So we have seen for yesterday also, Dr. Subradatta ma'am said, and global temperature is changing. All of us know, बहुत गर्मी लगती है, right? पसीना चलता है, बारिश नहीं होती है. So as you can see, it is unanimous, right? N number of different agencies here like NASA, HADCROT, NOAA, Japan, Berkeley, all are very agreeing. All over the world people are agreeing that yes, temperature is changing, right? So what? We have ACs, right? No problem. We can sit in central AC. The consequences of what is that? Not only if any of you think. So I also understand that some of you may not like to ask questions. I might get complicated in due course of the talk. Feel free to ask questions. At any point where you think, raise your hand. I will accommodate the question. So what happens? Will only the food crisis will happen? What will happen? So I just compiled few reports published in 2022 across the globe. There are hundreds of such reports, just few, and probably they are focused on Indian subcontinent based. Immediate effect is temperature rises, early apple flowering expected in Himachal. What will happen if the flower is early in apple? Food quality, apple quality is not going to be so great. It's no more as tasty, delicious, juicy as you like. India wheat production, exports wilt and heel. So you all know that Prime Minister Modi, Honorable Prime Minister Modi, first said that we will become the feeder to the world, but immediately we realized that actually our own production gone down by 15%. We had to stop the export. So falling rice acreage in eastern India is symptoms of a larger problem with environmental, equity, fiscal dimension. We'll start transplanting paddy early. UK plants flowering a month earlier due to climate change. Scary spring. Earlier blooms are a sign of climate change. Antarctica's only flowering plant have been growing more rapidly. So problem is all across. UK, Japan also, I think I did not put it. And the worst is a heat wave's lamented victim, the mango, India's king of fruits. Now, this is all not only about food. So it also affects the pollination. So there is no insects over there. So it also destroys, if there is no pollinators, so all across your ecosystem, all forest flowerings, like I think colors missing from city edge flowers on trees wither due to heat. This is from Bangalore. So tree ecosystem will be gone. So there will not be any other animals, birds over there, no pollination, nothing. We will on way probably to become a desert very soon. What will be the implication? Not only on food, but complete ecosystem. So it is under threat. So how it can be solved? Very difficult. But in the context of plant, what I can tell you is plant development is highly adaptable to biotic as well as abiotic factors. Like here, competitors are there, light is there, disease is there. They can't walk, right? We know. If we feel heat, we come here. But they can't walk. So they have to face all these challenges standing over there. Water availability, pest, and also temperature. So appropriate growth and development is required, which will probably manage the yield. So this is a very old report, 2009, that if we increase the temperature, if the climate change, due to climate change, if the temperature is going to be increased by 1 degree, we will probably lose 10% of the food grain. So, for your understanding, what are the symptoms we can figure out that if the plants are under some sort of temperature effect, some sort of, or drought or anything. So, leaf hypomesty. So, plants usually like to dance like this, So, that's very normal. If the temperature is low, they tend like this. But if the temperature is high, they go like this. So, you can see it here. Hypocotyle elongation. So 22, this is a very small hypocotyle. At 28, they grow quite fast. So this is the rosette, like biomass, or you can say the area covered. So all these things there is one transcription factor. This is a BHLH, all of you might remember, yesterday Dr. Subra also talked about importance of BHLH transcription factor. This is called PIF, phytochrome interacting factor. And phytochrome is a red light receptor. So this somehow integrates all these informations, transcribe this for certain gene regulatory mechanism, and helped by one plant hormone, growth hormone, that is called auxin, and somehow it gets modulated. Now, is it so easy? So some of you must be knowing Paul Nurse. He was a Nobel laureate. He is a Nobel laureate. I have pleasure to meet him a couple of times because he was in the advisory board of Sinsway Laboratory, Cambridge. So at least he used to come twice in a year. So he got the Nobel Prize in cyclins. So you will also see, I also worked, as I showed you, Embo Reports paper, where we showed cyclins. So Tim Hunt and Paul Nurse got the Nobel Prize for discovery of cyclin genes. So life, logic, and information. Focusing on information flow will help us to understand better how cells and organism works. And this is really crazy. We don't know. Right, as you can see, this organism, so many circuits, that too in different states. You try to fix one, others gone. Right, like, all of us see this in Noida. इतना voltage fluctuate होता रहता है. एक fuse ठीक करो, दूसरा उड़ गया. Press लगाओ, सारी light बंद हो गई. Right? So this is how the life also works. खाना खाया, acidity हो गई. अब आप सोच रहे हैं भूख मिट गई, अब कुछ कर नहीं सकते. क्लास में यहाँ सुन रहे हैं, कुछ को नींद आ सकती है. So all these things are to make you awake, right? I can see a lot of students. So how do we do that? So all of you know that if you go in light under the sun, you not only see the light, but you also feel the heat, right? That is the temperature. So light and temperature is almost integrated. So whenever plant or human, so some of you might be knowing the last year's Nobel Prize for the heat sensing mechanism. And the system was chili, right? It burns. So the Nobel Prize was meant to discover the receptor on the test bud, which gives the signal that you feel the heat caused by the protein of the chili. So such kind of sensors are in plants as well. We are yet to discover those though. So these two signals, light and temperature, go together as an input, meets the sensor, and then pass on the information through all these complicated networks. And what is the output? If the temperature is low, you get a very nice hyponasty plant. If it is high, you get the elongated. So germination, growth, flowering, etc. My scientific contribution has been in different ways. So I identified the master regulator of temperature sensor in plant to develop climate resilient. It is ongoing work, and some of them are there, you can see. So phytochrome function as thermosensors in Arabidopsis, chloroplast signaling gates thermotolerance in, these are 16 and 18 paper. At the same time, I will show you that it is light in terms. So all of you must be knowing the red light receptor is phytochrome, blue light receptor is cryptochrome, UV light has another receptor called UV8. So if you know that reports are already there, the cryptochrome directly interacts with PIF, all are converging on PRIP4, which I showed you, the master transcriptional regulator for growth in plants. So this is a complex network. And as you can see, even if there is no phytochrome in plants, if we generate a null mutant, this is the figure from science paper, phytochrome null mutant ABCD, there are 5 phytochromes in plant, particularly Arabidopsis. In rice, there are only 3, ABC. So 5 ABCD, plants are happy. So that simply means that other receptors are compensating that. So I blurred it, but now my studies with the chip-seq analysis, I am not putting all these, how do we discover, but we discovered, so if there is no phytochrome, so if there is phytochrome, as you can see, they inhibit PIF. So there is no growth. Like this is control plant. You can see it is not growing much because phytochrome is there, which is inhibiting the PIF, so plants are not growing. But as soon as all phytochrome is not here, this is not there, PIF is there, and it activates MPK3 and the growth is happily happening. So now we have identified which is the gene or factor which is responsible for the growth. This brings to a very fundamental question. What is required for growth? Energy, right? If you go for shopping, right? What is required for shopping? Money. If you don't have money, you can't do shop. So in the cell, money is ATP, right? So we discovered that this is the plant growth. For plant growth, money is ATP, and if that particular thing which regulates this availability of ATP is not there, probably PIF4 can't make it grow. So that's it's fairly proven with that. We are still working. Now in the another aspect of the same work, now you all know that if one plant is flowering, even your plant which is in-house or far away, inside, everything is flowering. How is it happening? We all know that, fine, आज की class नहीं होगी, WhatsApp group में डाल दिया, plants के पास नहीं है. What are they doing? Although they are certain, like flowers have some sense of, secondary metabolites. All of you must have heard hing. Some of you might like it. If you open it, you smell it. So plants have some sort of those chemicals. I call them plant communicators. So what I identified, that there is some mutant plant which perceives the temperature even at low temperature and making the plants grow like this. I am not revealing the complete name here, but this is terpene synthase, one of the terpene synthase, and I already mapped it using next generation sequencing. So we know the gene precisely, which gene is responsible. So you can see that these plants can flower even at low temperature. So where is it expressed? So we all know that has to be expressed somewhere, right? Ideally in flower, because flowers are the source where you have nice smell, right? So once the flower is there, there is some smell. So as you saw earlier, if the temperature going up, flowering, plant flowering is becoming early. And as soon as a single plant flower, it starts emitting this secondary metabolite. So that nearby everyone knows, guys, ready for the reproduction. It's time. Okay, make flowers, make seeds, because temperature is going to be changed and it can affect the fitness of these plants. So this is there, precisely there. So what happens after flowering? Seed. Seed is formed. So this space is missing because there was a confocal image, a nice image. But anyway, I will explain it here. So this is from a very recent EMBO report paper. So for seed growth, we have this embryo. Seed has three components, all of you must be knowing: embryo, endosperm, and a maternal seed coat, which exclusively coming from the mother side. Right? Embryo is the mixture of father and mother, same as endosperm, because of a very nice event called double fertilization. Right? One sperm meets the one egg cell and second to the central cell. Right? So central cell fertilization makes your endosperm. Right? Egg cell fertilization makes this embryo. And for the proper development of seed, a proper coordination between all three is highly required. Now, how does this happen? This is a dicot model. In monocot, it is slightly different. But I have kept this image as a in between. So as you can see, the embryo is only up to here. In monocot, embryo is just only until here, but in dicot, what happens that this whole covers, the embryo covers everything. Right? That's why when you germinate the seed, two cotyledons come out. But if you germinate the rice seed, it is not like that. So this is although another picture. This is also a confocal picture with the fullerene staining, which shows that very nice, precise endosperm distribution throughout the seed. But in cyclin mutants, we have very large endosperm nuclei, no cellulization. That's why the seeds are very small. And that happens because of the disturbances in microtubule orientation, which was here, somehow not coming. Now, we got the seed. खा लेंगे कुछ भी मिलेगा तो. What we want? So it again, as Dr. Ankush said, tuberculosis happens in low income area. Same thing, अगर हम low income area में हैं, बस्तर में है, जो मिलता है खा लेते हैं. यहाँ नहीं खाएंगे. What we need? Nutritious diet. Okay? How to do that? Because there is no one. Plants, they work for themselves, not to feed us. Right? It is us who select. That is called the process called domestication. Right? Wild rice or wild wheat, these are not meant for us to feed that, okay, human population is going to be so many million, billion, and we need to take care of humans. No, it is not like that. What humans did, they started selecting the varieties or species which could be beneficial for human. And this whole process is called as domestication. Right? So here we need to do some sort of domestication, and all these works involve genome engineering. So what I am showing you here are the rice varieties, slightly hidden numbers, but rich in iron and zinc, and also some sort of cellular based required heavy metal that is called manganese. So we will talk about it later, maybe some other time, some other conference. So iron and zinc is highly required. Where? For developing fetus at the very early stage of human development. Some of you might be knowing that during pregnancy, in India, it is strongly advised to consume iron tablets, right? Why? Because we lack iron in our diet, and iron is absolute necessity for developing fetus. So what you can see here, and in general, rice has hardly 10 ppm or milligram per kg seeds iron or zinc, hardly. So the rice varieties we consume, there no iron and zinc. So, and some of you might have heard about this golden rice with vitamin A, etc. So, but these were with the help of other technologies, but that shows that it is required. So here, there are varieties which are very high amount of iron. For instance, this one 150, ppm. mg per kg is ppm, both are equivalent. At the same time, very high zinc, like this one, and some rice which has both in high, some are both in contrasting features. Now the obvious question is, why am I highlighting those? As you saw earlier, that life and process is very complex. So if I just focus on one, which is having very high zinc or very high iron, there is a possibility that I might not find anything. So I selected the rice varieties which are having both in excess and having one in excess, another counterpart in low. And we call it genetic dissection. Okay? So one trait, another trait, we cross, right? But here what I am doing is identifying the genes and editing them. So similar story we published in Trends last year. So at the same time, we are also looking on potassium. This project is about to finish. It was a collaborative project with Delhi University South Campus. There is some biochemistry over there, like hyperactive kinase, constitutively active kinase. Some of you, if you know, it is good. If you don't know, you can ask. And the next thing is that we came to know that there is some traits known here, right? But how about how to find out something new, novel? So we do this through a process called genetic screen. Okay, so we developed a population of rice mutagenized by a mutagen called EMS. Okay, which creates a single mutation, C2A, and leads to lot of trait. So what you see here, this work is being done in AIG, in MET. Some of, like Dr. Naveen is aware of it. So we grow these rice lines. So there is a lot of innovation here to see. So we have huge number of lines. So do you think 5,000 rice, how much? You may need 1 acre of place. Here there is 13,000 of rice lines are growing in these few trays. So what we did, we used 96 well PCR plate, spent whole day cutting their tail from the bottom, cleaning them, putting each seeds there, and you can see these are growing. So one tray, we are growing 800 rice plants. One tray. Now what's the advantage of it? We can see the roots directly there, how they are growing. We can see the shoot, and we can alter the nutrients provided to this medium. Like, if we don't want iron in it, we don't give them. It is in my control how much I need to feed. It's like my baby. If I want to feed very nice, healthy cocoa water or Nutella or something, that's in my hand. So I can play with it, right? So my students are playing with it. I usually don't go in that. So here, this is very nice picture. You can see the rice growing, and you can see the roots. So what's the outcome? You can see here different, this is the control. So you can see, I'm not sure, yeah, it's fairly visible. So there are two traits you can see easily. There are multiple traits though, hidden, and I will not take much time in that. So root length. So if you see there, these are the very clear here where my cursor is, you can see the seed. So root is here. But if you come here in this line, which is mutagenized line, root is almost 3 times longer. Now what's the advantage of longer root for rice? It's highly desired, because rice is a crop which is really bad for our ecosystem. It drains water. Millions of metric ton of water goes every year for cultivating the delicious basmati rice we consume. Almost water level is gone in Punjab. Okay, so if there is longer roots, what will happen? We need to give less water, because roots can go down. And that's why some of you, if you might have gone to any field, you might have seen that rice plants are kind of immersed in water. Why? Because their roots are too small. They need to keep water almost ready for them. At the same time, now second trait, what you can see, anyone can raise the hand if you see any second trait here. Longer plants. So you can see this is 12 centimeter stick, white stick, okay, which not only serves as an identification tag, all have their identification on that white tag, but also is a measuring tape. So we use another computational tool called Fiji or Images. Some of you might be knowing. We go there, we put this image, we mark that this white is 12 centimeter, and then we measure the root length or shoot length. So here you can see this line is fairly long, okay? And these are not that. Yeah. And some other things are also important. So flag leaf, you can see these are tilted at certain angle. Why this is important, I will come into the next slide. So what's the general root length, what you can see? So these are 542 lines. All the root lengths are measured with the mean value, etc. So Nagina 22, which is a mother plant here, mother variety, it has 8 centimeter root. Now you can see we have roots as long as 22 centimeter. Right? So basically we have approximately 200 lines which have much longer roots. And very few lines also there which are less than 6 centimeter. So these we will see how, because plant development we might need to know these as well. So this is like one example here for you. So as I said, in the root what happens, other traits what I was mentioning, this is the paper from my previous postdoc group. So you can see that generally all plants have nodes and internodes. So nodes and internodes are there. So what are nodes and internodes? From where your leaf or branch emerge. So wild type you have this very, very nice phyllotaxy, like one side, another side, another side. And there is one mutant called Lux. That is an evening complex. That is the complex of genes which determines when the sun is set. Okay, so there are 4 or 5 genes in that. Elongation factor 1, ELF3, Lux, time of coffee talk. So if you mutate this, what you will see that this kind of node and internode pattern is changed. And you can see that there is almost no elongation of nodes, right? So these kind of mutants also emerged over there. What next? The first thing is to control flowering. That's my message coming back. So I started with root length, plant growth, then flowering, and coming back to seed. So we need a system where we can tune the plant growth fairly. So this is what I am showing you. Almost all of you must be knowing that this is not the time for rice flowering. People are sowing it, transplanting it. I have just visited Kolkata a couple of days ago in our field where these plants are growing, and we transplanted rice last week. You all are invited to see flowering, seed setting in rice in AIG laboratory. So we developed an excellent system where we can grow rice throughout the year till flowering stage. Why this is important for us? Because we want to develop a fast breeding system, that we develop new varieties by crossing. Breeding is crossing. You take one variety, you take another contrasting figure, cross them, as I said, mother and father plant, pollinate them, and you have traits. Screen them. In 1 year, rice usually is grown only once in the field. Here, if you go to Cuttack in Odisha, you can grow twice. But here in this facility, this rice variety is flowering in 60 days. That is 2 months. So theoretically, you can grow six cycles of this rice in AIG laboratory. So basically, in 6 generations, you have a new variety of rice. 1 year, new variety. So this Jink site or something, we aim to develop new varieties within a couple of years. Tested, all sort of analysis done in 1 year. So how do we do that? We also have all sort of around 50 different genome editing cassettes for monocots, dicots, Arabidopsis, rice, wheat, all sort of. Plasmids are available, methods are standardized, and this kind of system. So our lab is like unique place that you go to lab, then enter into plant growth facility, take your sample, take it out, do the PCR, qPCR, everything, put your sample in -80, everything in one. And tissue culture is next door. If you want to transform something, do it all in one room. J3314. Any of you are most welcome there. So the work is mostly done by these 3 students. 2 of them are MTech students from AIB. So collaborations list are there. a lot. So I will not take, as I said, I have only 20 slides. I hope I didn't take much time. But before I say it's done, I must thank our Honorable Founder, sir, to provide kind of setting up all kind of financial support to make this lab practical where it is. Then I have a couple of projects from ICMR, DST. So all these fundings are there. So I end up here that all of you are welcome. We have PhD positions open, and obviously the MSc admission is open. Please remember, only 10 seats. So I look forward, the brightest of students coming there, do practice the cutting edge, most cutting edge technology. We do RNA-seq routinely. I didn't include it here because it was more towards genome engineering. We do chip-seq in the lab, a lot of bioinformatics, high-end data analysis. So if any of you are interested, welcome to visit there, J3314. I sit in J3307, so anytime. So any questions? No? Did I speak too bad? Now I can ask. You know, I am faculty here. So now it is exam time. I can ask any of you. What did you understand? Who would like to answer? Oh, no. Chalo, I know that all of you must be very hungry. Thank you for your patience. Thank you so much. Once again, sir, thank you for inviting. And it was a pleasure talking to all of you. Thank you so much.
Thank you very much, sir, for your wonderful presentation. Now I would request Dr. Aarti Goyal ma'am to felicitate, sir. Thank you, ma'am. Now I request all the esteemed dignitaries, delegates, and all the attendees to proceed for networking lunch. We will meet after 30 minutes at 2:30 p.m. Thank you.
Welcome back, everyone. We'll now proceed towards the third session planned for today.
Our next speaker is Dr. B.N. Malik, Director, Amity Institute of Neuropsychology and Neurosciences, Amity University, Uttar Pradesh. After completing his PhD from AIIMS, he joined the School of Life Sciences, JNU, as Assistant Professor. And since 2001, he continues there as Professor of Neurobiology. He held several positions as member chair of several national and university level committees, including Dean Life Sciences JNU during 2013 to 14. He was the first person in our country to initiate REM sleep research. Post superannuation from JNU, presently he is Director of Amity Institute of Neuropsychology and Neurosciences. His research focuses on understanding neural regulation and functions of REM sleep. He has published more than 100 research papers and book chapters and has edited 4 books. Dr. B.N. Malik has been elected fellow of all the 3 nationally recognized academies. He has received several national awards and recognitions, including Shanti Swarup Bhatnagar Award, National Bioscience Award, Shakuntala Amirchand Award, B.K. Anand Award, and J.C. Bose National Fellowship. Dr. B.N. Malik's lab has been working on understanding the neurophysiological and neurochemical mechanism of regulation of rapid eye movement sleep or REM sleep. I would now like to invite Dr. B.N. Malik to deliver a talk on modulation of gene expression to understand behavior regulation. Over to you, sir.
Thank you. Okay, good evening, everybody. Thanks to the organizers, particularly Professor Verma. Rather, Verma ji, I know him since 1986, ever since I joined JNU Life Sciences. Thanks to the introducer, I don't know his name, but thank you for the kind introduction. Well, coming to the topic, yes, as you have heard that I work on neurobiology and particularly neurobiology of REM sleep. So I guess most of you, if not all, are unaware of this, what is REM sleep. But I have to first introduce that to you. And how come I am here? See, I am not by training as a molecular biologist, as you can understand, those who have passed out in mid-70s, hardly we knew about gene regulation and much other things. But one thing is true, if you have to remain in cutting-edge science, you have to keep yourself abreast. That is what we have been doing. So as such, I may not be knowing the fundamentals, but I have used gene regulation. And that too in a model system of cognitive science, which is probably one of the things for present century. And as I went through the program, quite often, majority of them, you have dealt with in this conference. This is a colloquium on emerging trends in genome editing, and majority of them have dealt with the plant system or microbial system. Here I will try to enlighten you a little bit on the cognitive part and how it has been done. Of course, I have not directly dealt with the gene editing as such, but its product through the protein by modulating their gene regulation through siRNA or shRNA. By using that, what we have shown, even in behavior, that by modulating the neurotransmitter synthesis, that you can modulate the behavior as such, and its fallout different disorder symptoms. So we have reached up to that level. The next step is to use it as a practice in medicine or go as a development or any other. So by the time we finished my career in JNU after serving 36 years, we had reached up to that phase.
And it can be picked up by anybody or none, whatever. So I will start from there. First thing, I will introduce the subject and how it has been used for this mechanism. Okay, this is a brief outline in the next 35-40 minutes. Therefore, when Professor Verma asked me, I cannot say that he requested me. I have to obey his orders, right? I did tell him that I have not worked as a molecular biologist or molecular genomics, gene editing, but he said, no, you have to talk. So I said, okay, let me talk on this. So I have given the title of this presentation, Modulation of Gene Expression to Understand Behavior Regulation. So now let me take you, I'm told that I cannot move from here because otherwise it will not get recorded. But usually my habit is to walk around with a collar mic, but that is not possible here because when you speak, your body language also speaks a lot for making a sense. So here, I must tell you, Watson Crick, most of you have heard the name of as a Nobel laureate, what he had said. There is no scientific study more vital to man than the study of one's own brain, because our understanding of the entire universe depends on the same brain. And based on that, we are trying to understand the same brain, how much we can do. So that is the essence. And the point is, it's a complexity more on that to understand the behavior regulated by the brain. And therefore, if you go through the literature, there is no demarcation who is a neuroscientist. Of course, the neuroscientist is there, but to understand the subject, you name anybody, be it from neuroscience to mathematician to psychologist, everybody has jumped into the fray to understand the brain. This is, to put it most simply, this is a simplified diagram of a brain, if you may say so. Well, it might look like anything, even at a synthetic biology, if somebody is trying to design these days. This is more complex than that, cell within a remarkable shape and size. Here you're talking about the entire body, including the brain. And beyond that, what is a thought process? And we are trying to understand that. Amazing. I'm giving you another example. This is a bird which is named as Zarzur. You know, the way these are, and the physicists and mathematicians have calculated, these birds, they must be calculating 100 million calculations per second so that they do not collide with each other. And what is this brain that is doing, and that is being done at this type of connectivities? Okay? With this background, on one hand, my research focus was to understand REM sleep, that is rapid eye movement sleep, and that is that stage of sleep when most of us dream, or majority of the dreams occur. When I was a student like you, the textbooks hardly used to have names, 1 or 2 lines, and whole sleep used to be half a page or 1 page. REM sleep was just 1 or 2 lines. Well, that was one of the reasons, and fortunately I was introduced to the subject and I wanted. And yes, I started REM sleep research in this country first, of course in animal studies, and that is in JNU. And fortunately these people selected me to join there. So once I joined there, this is some example you might have seen it many times. People knew these things. See, in research, what do we do? It is existing. We are trying to uncover. And therefore, when we try to uncover, there is a lot of emphasis on the existing and contemporary knowledge and technology. So, what I have studied in 70s as a student, early 80s as a student, if you know only that much, then you are already my age. That cannot happen. You have to use me as a stepping stone and excel beyond me. That is what I feel as a teacher. So these phenomena, what I showed you, many of us have seen. Now here on your left, the gentleman who is Aserinsky, and on your extreme right is his daughter. It is a prized possession picture that I could get. One of my friends shot it, in center obviously me. This is 1995 Nashville meeting, and this gentleman had discovered this REM sleep as an experimental. He himself was not PhD, but he was working with somebody and he discovered this. Though it was done on many of the students, but his daughter, she also had some contribution to it. Okay. Now, what is REM sleep? Rapid eye movement. You can see the eyes are moving here. Well, sorry. So eyes are moving here, we will find. Here for the baby and here for the dog. This is there in different species. And so far what we know of, it is existing in all higher animals. And more we are gathering information. We are getting it in different animals as well. But we do not know how to interpret, how to record that. Because of smallness, and the brain is not that evolved. Just 2 days back, somebody sent me that probably the spider also has REM sleep. That report has come in Washington Post. The science was done in Australian scientists. For rat, dog, cat, all certainly we know these are there, but we do not know what dream they dream. But that is true with all human beings. If I dream something, if I don't tell you I am dreaming my mother or a cat, you would not know. So that's subjectivity anyway there. But dream is there. So science, we decide on that whether dream is there or not. I was interested on which part of the brain, how does the neuron communicate to bring in the dream. What dream? Even today we do not know. The science doesn't know. And second thing, if its disorder is there, how does it work and what should be the mechanism? That was my topic of understanding. Does it work in animals? Yes. So we have shown, so this is what is happening, eye movement, and this is how we record electrical signaling. Therefore, you can model it in animal and study. Okay. This is in the cat in my lab recording, and this is in the rat by different students whosoever has done. Just to give you a glimpse, this is what we record. Again, we are doing REM sleep when dreams occur. That doesn't mean I am working on dreams. So please do not ask me question later on, because at the end of many of these talks, people will say, oh, I have dreamt this and that. What does it mean? Well, I may not know. Even if I know, I may not be able to, I shouldn't be able to tell you to express my ignorance. But yes, I know the neural mechanism of that stage of sleep. This is there, as I said, in different species. Fine. So this is one aspect. These are existing. What will be the neural mechanism? Also, it was known behaviorally these things happen. And that is nothing new. Even 100 years back, people knew that if you don't sleep, what happens? Problem. So I would like to emphasize certain part that so far, evolutionally, we know that it is there existing even million years ago. It is reduced with age, increase in babies. REM sleep is reduced in all these disorders that is there, Parkinson's, Alzheimer's, psychological mood disorder, even simple fever and all. And some fever it will increase. And in certain times it is increased, like narcolepsy, depression. Then, if you experimentally do, it has been shown that if you causes of REM sleep, if you deprive REM sleep, anything else? Okay. So if there is a loss of REM sleep, there are certain behavioral expression like irritability. You get very irritated. That quite often many of us have seen. For example, if you have gone, if somebody is sick at home and you are awake whole night, or you have traveled through train, or before the exam, and next day when you go to college, school, or in a family matters, you get irritated and behave in a manner you are not supposed to behave. You repent later on. One of the major causes is sleep loss. One of the major causes of sleep loss is aggressiveness. then confusion, concentration loss, and that is one of the major causes of road transport, air, industry accident. So this. Similarly, if there are loss of REM sleep, the brain development is affected. That was also known. Memory loss is also there. So having said this, what I proposed in late 80s when I started my career, therefore REM sleep cannot be of vestigial origin. If it was there, in millions of years evolutionary it will last, is not serving any purpose, it is not possible. We did not know the mechanism, nothing was known. We knew only this evidence that this happens. If it is so, what type of fundamental function it may be doing in the brain? That was my question. And how is it doing? Okay, there are many other reasons why I thought of. With my own maturity and getting more data, we have given something, but fundamentally this was the thing. If that is there, what I thought of, if at all I have to understand how REM sleep loss is affecting, what should happen? Like if I am standing here, this pointer is not working, then unless I know how does the pointer work, what should be the mechanism? If I did not know that it works with a battery, I will not even think of. So if I knew that, then I will try to change the battery or the bulb or something. So we needed to know the mechanism. Now, as I said, when I started my career, the REM sleep was discovered in mid-50s, 1950s. I am born in 56. I started in 86. So it was, and in the textbook those days, it was just 1 or 2 lines about REM sleep. So not much was known. So I thought then first thing I have to understand the mechanism. Not that nothing was known, very preliminary things were known. That was these 2 types of neurons, the black type and the green type. That is just for depiction. The black is noradrenergic, green is cholinergic. For some reason, I did not agree on this concept, whatever that was given in 74 and 80. So what I proposed, that should be with the red one in between. And we proved it. All right, that is the GABA one. Fair enough. Then we proved there should be other connections here. All these are work done in JNU by different students. They are in their own capacity. Somebody is professor in UCLA, somebody is a faculty in Harvard Medical School, somebody in Germany, all these places there, even including in India also several places, people are there. Then we worked about neurotransmitter. Each of them is a thesis, by the way. So if I am talking about these, each slide is 5 man-years work, which I will not, but then I have to bring home certain points, therefore I have to give you this background. So we talked about different neurotransmitter, this is different receptors. This is important because based on that only the gene regulation will come. And this is how the mechanism, and ultimately this is what has come, and this is a cover page of Progress in Neurobiology, is one of the best journals, and nobody can understand REM sleep without understanding this type of neural connections. So this is what is a contribution, what we have done here. Fair enough. Now what is it? Here that means some of these neurons, as you can see here, these neurons, they are continuously active normally when somebody is not sleeping or not REM sleep. Therefore it is written here awake. During awake, these neurons are more active. During non-REM sleep, see, those neurons are not active, but these become active. And then during REM sleep, see, this is totally silent. Some other neurons are getting active. Means what? In the brain, it is not all the neurons are always active. There are specificities. So if that be so, what we have understood, to put it simply, this type of neuron, which is most active during all other stages, they shut down during REM sleep. And if there is a REM disorder or non-REM sleep, or even not allowing REM sleep, those neurons keep on being active. That also is my work. Means what? What do we expect? This is what it is as a diagram. This is Can you hear now? I don't know. ये ठीक है मैं ऐसे बात करूं because I have to show this. See, if this is a neuron, the black one, this neuron is always active when somebody is non-in REM, and it is releasing neurotransmitter, that red one, and that is noradrenaline. Normally during REM sleep, this neuron stops. That means this release will not be there. But during deprivation, these neurons keep on firing. Means what? In the brain, there should be more flooding of this neurotransmitter. That much I think you understand. If that is so, then if at all I have to understand the disorder, one simple way to think, that means disorder may be because of this neurotransmitter. And I have to prove that. So first thing to prove whether is it the neurotransmitter actually is increasing in the brain? Yes, indeed, we did HPLC, we took out the brain, we did the neurotransmitter or the CSF, and we did, yes, it is increasing in most part of the brain except this hippocampus, which I am not going at this moment, but most part it is increasing. Now, if this is increasing, and we talked about, if you remember I showed there it is confusion, excitability, memory loss. Do we find memory loss in a liver cell or a muscle cell? We are talking all in the brain, and reactivity in the brain in the neuron. Then what should be the difference in a brain cell and a muscle cell and a liver cell? Primary difference is excitability, electrical signaling, which others are not there. So if that be so, then this neuron should be modulating, this neurotransmitter should be modulating that excitability, because after deprivation one is losing memory, many things, at least temporarily as an initial stage. And what affects the excitability? Among many things, the primary factor is sodium potassium ATPase. So in 90s, late 80s, 91, rather 88, 89, first I proposed that there should be that deprivation should affect neuronal excitability by modulating sodium potassium ATPase. And indeed we showed that it does affect sodium potassium ATPase, and then over the years, as I said, each slide is my one thesis. So we showed how it modulates. We went through all the stepwise manner. Then we showed how this is working. But if it is an allosteric modulation of the enzyme, this is temporary, short term. How will it work for long term? Indeed, it is transcriptional, translational modulation of sodium potassium ATPase, and that could lead for long-term excitability or behavioral changes. And that is again because of those noradrenaline increase. All right, so I am proving up to that point. So I have said these are the effects, and when we are talking about these effects, so far I talked about excitability, short-term effect. As I said, if I have not slept for 1 or 2 nights, I have traveled, I get irritated. So these are short-term. But with loss of memory and all, these are long-term as well. Therefore, there should be long-term deficiencies, even brain development. As I showed you, one of them is brain maturation deficiency as well as memory. So if that be so, here the evidence. After REM sleep loss, these are quite tedious, hard studies, in the sense you are depriving the animal, then taking out the brain. You are tracking the neuron for about 250, 300 micrometer, and see the neuron are having these branches. And you see in the deprived one, I don't think you need a great computational eye to see. Here the branches are much less as compared to the normal. And if these branches are less, the synapses are less, the memory is lost. This is the first time we showed that this is happening. Each of these neurons tracing takes 6 to 10 hours continuous tracing under microscope. Okay. So these were done by Shatrunjay Giri and others. Then these are electron microscopic, where you are showing even at the synaptic junction, here the length of active zone is increasing, not the synaptic cleft size. Cleft size is not increasing, that means well development. a neuron has come and getting connected. The space between this is not changing, but size is changing, and that will modulate your behavior. And that will modulate the neurotransmitter quantum release. So that is what we have shown. And not only that, here the pre and postsynaptic proteins, monk protein and the postsynaptic density protein here, PSD95, monk 18, they are also reduced. That means these are structural changes this way. So not only neuronal structural changes, they are synaptic this as well. I will not show this, but we have gone ahead. We had done primary culture. We have developed how the neuron is growing and making a synapse. And then we have shown how the neuronal growth also getting affected by noradrenaline, and how is it modulating them for the development of the brain. But at this moment, I will not show all this. Now, I think I have gone almost half an hour. I have another 15 minutes in 45 minutes. So I will now switch to, if these are known, how do I do with the topic of today's lecture of the gene regulation? So if we have talked about these noradrenaline because of this neuron here, which is releasing neurotransmitter, if deprivation associated effects are mediated by this neurotransmitter, which earlier we have shown by receptor blocker. But look here, if a neuron is releasing neurotransmitter that is all over the brain, now if I am blocking the receptor, which area receptor I will block? I will show you the overall in the brain. I have to show specifically what is happening, and that too in a systemic long-term manner. Inhibition of synthesis of noradrenaline should prevent loss associated effects and should affect REM sleep as well. Now, if I have to do that, noradrenaline as such is not a protein. And if it has to be a gene regulation, then how will it be doing? Gene regulation is affecting through mRNA some protein only. So this is what I have depicted here, how the neuron is connecting through neurotransmitter. And if we see any cell signaling, a cell communicates with another by releasing some protein, and in that protein or proteinous body or glycosylator or whichever post-translational modification, there will be change in the gene regulation. And that way it can modulate, which is I was trying to say. Now, if the gene regulation is there, even if you change the gene, all of us understand there is a big role of epigenetic modulation. Even if you have a transcriptional difference, now two things, either transcription might get, even if the genome is different, if the epigenetic modulation is not there, you might not get that protein, or there may be any other modification, or even if the protein is synthesized, there may be post-translational change. All these can happen. But then what is essence here? Essence is ultimately you have to affect the protein part which is modulating, whichever way you do. So this is how mRNA regulation. Therefore, this gene regulation can do, this is what you must have heard in different lectures, so I am not spending time. But epigenetic modulation is coming in a rigorous way, in a more emphatic way. But yes, every system has its own advantage, disadvantage. If you want even to change at the transcriptional level, it's a transgenic level, next generation and all, by all means you do it. But if you have to modify as a support disorder, if you try to do at the gene level by changing its genomic, then probably you don't know what you might land up. But suppose you are doing at a cancer level, though I am not an expert, but if you have some mutation has already taken place, then probably it will be worth. So it depends on what you want to do. My purpose was, if there is a disorder at that level, should I go at the gene level? And even if it is there, what should I do? Therefore, I preferred to do at the transcription level. And as I said, there are epigenetic modulation. Yes, even deprivation modulates epigenetic level by methylation, acetylation, and thereby modulating their transcription. We have shown that. And this is what is a classical book or internet somewhere. All of you must be knowing this, and you must have been told why siRNA or shRNA can modulate the transcription at this level and can modulate the regulation. But the point we have to understand, we are trying to understand here about the noradrenaline or norepinephrine modulation, which is not a protein. Then what do? Then you have to see here, in order to synthesize noradrenaline, you need different steps of enzymes, and the first step is tyrosine hydroxylase. And this step is a rate limiting step. So by modulating that, I can control the synthesis. And noradrenaline evolutionarily existing even in the lower species. Epipramine at the lower level, it has a cyclic ring there. But that is the chemistry of it, which at this moment I am not interested. I am interested how to modulate its synthesis. What we did then, we used siRNA or shRNA of tyrosine hydroxylase so that I can modulate noradrenaline synthesis. And if I can modulate that, this is what is written. Hypothesis was, if noradrenaline synthesis is tweaked, as in different disorder, mind it, I have to do it in a behaving animal, then REM sleep should be affected. And if REM sleep should be affected, rather if it is reduced or increased, by adding noradrenaline, I should bring back behavior. Mind it, we are doing even at the cognitive level. And this is most complex behavior. And once that is there, then if REM sleep reduced is there, all the associated symptoms, like I said, excitability, I said neuronal damage or apoptosis, which we have shown independently, all those should happen. And if I can bring it back, I should bring back all the behavior. Indeed, we have done this. Now, this is what you can see here. This is what in the animal here. In this animal, for standardization, we did standardize even in cell culture, but which method to be used for injection or for moving or injecting the siRNA and shRNA into the cell outside. Then we did it in the animal. And see the control here. In one animal, means rat, here, this is histology. On one side of the brain of this locus coeruleus, where these neurons are there, we are injecting siRNA in one set of animal. In the same animal, on the other side of the brain, this side, we have injected the control one. Similarly, in other groups, we have used shRNA. Then, this is a control. Then we have punched out these areas. Punching out these areas means these are millimeter or half. Then you take out, then you do Western, and then without punching also you did histology. You can see here, this is histology. This side is control shRNA, means scrambled RNA, and this side is shRNA. Similarly with siRNA. You can see with tyrosine hydroxylase immunostain, this side, here you have so many neurons still there. This side is not there. And here are the counts. After shRNA, you are reducing by about 70%. And if you did Western, you find here it is reducing at this, but in control you do not get that. Means what? If you do this, that means you are reducing tyrosine hydroxylase synthesis. And if you reduce tyrosine hydroxylase synthesis, this is a rate limiting step enzyme, so it will not allow noradrenaline synthesis. What we did after siRNA or for shRNA, we waited for 5 weeks, and we did again in the same animal and shows that it is coming back. So we confirmed that. Now the behavior. Yes, ATPase activity, if you can see here, this is 1993 study. That is First time what I showed that after deprivation, ATPase activity increases. Look here, it is increasing by about 60%. 10 years later, 2003, what we did, another type of experiment by inhibiting those neurons, again 60-70% increase. And now, by withdrawing noradrenaline release, again about 60% increase. That means this is the thing which is happening on ATPase. Now, how about behavior? Very complex slide, but then you have to see the entire behavior. Normally, this is happening, noradrenaline is released at this area in a normal REM sleep. Here, this is normal REM sleep. Here, I have injected siRNA or shRNA. Means what? Noradrenaline, if you see here, depiction, much released. If that is there, there was increased REM sleep. In these animals, if from external source, if I am injecting noradrenaline, REM sleep is coming back. And in these animals, if I am depriving of animal of REM sleep, ATPase activity is not increasing. Means the symptom of REM sleep loss, what many patients are getting is because of these neurons, this noradrenaline. Now it is from bench to bedside. That is what some people have to take it forward. From nowhere, from almost zero, we have come up to this level. Of course, it took maybe 15 theses, so 15 into 575 man years, and then so many years to come up with, but this is at this level. Then how was the neuronal damage? We had shown that this is through intrinsic pathway by the apoptotic modulation. But if we are inhibiting this noradrenaline synthesis, that is also not taking place. The word you should see here, caspase 8 did not get effect, but rest others. So that means internal pathway is working for apoptosis. Now, if that is there, the next step should be, I have finished my innings in JNU, but this corona period, 2.5 years, I thought I will finish up to this stage. So we started with even zebrafish. This is what is a development of zebrafish. The idea was for me, by the time I had set it up, that now at this zygote stage, or at certain stage, if I can knock down or conditionally knock down these stages, do we get that type of behavior or something, which I could not finish there. Here I am here for 12 months. I finished 12 months, my entry here last week. All these are set up here now. I do not know how much I will be able to do, but any of you can pick up or none of you can pick up, or you can spread the word. This is how things can be done and the study can be extended. So with this, I will finish my talk and I will love to take it, but then I will leave it with this. After spending about 3.5 decades, it's written here, you cannot see where whatever the green part, is it sunrise or sunset? Whatever we have led with, and there's a long way to go. So science moves in this way. Important thing about gene regulation and all, what you people are reading in the textbook and all, hardly we knew anything in early 70s or so when we are starting. You have to keep abreast. This is primarily for people who are sitting at the back. But if you are knowing this, whatever is going on now, don't think after 10 years, 15 years, 20 years, the science will remain that. You have to keep yourself abreast. And that is fun of science doing. And with this, I will end my talk. It's my pleasant duty to acknowledge all the students, of course, other staff and other colleagues like Professor Verma and many others in the department who allowed me to work. But here on the left side, all these people who are directly involved and finished their PhDs and wherever they are located, including full professor is Noor Alam in UCLA, then Mahesh Thakkar is they are full professors, and like Sushil Jha and all who are in India and JNU and maybe UP, Patna University, Allahabad University, the faculty whosoever has been there. There are 2 or 3 more students whose PhD is yet to be done. They would have finished, but then because of Corona got delayed. By this year end, by mid next year, they will also finish. Some of the postdocs, 2 of them are written here. Rachna, who is here in AINN for last 6-7 years, ever since this department is there. And funding, mind it, all the funding comes from taxpayers' money, at least in this country. So I'm grateful to all taxpayers who have allowed me to work. And last but not the least, I'm grateful to all the animal lives who have sacrificed, who have given their lives for human benefit. See, whatever research we do, be it here or space research or sea research or life science research or agriculture research, sometimes we think, are we selfish that we are only doing for human benefit? But we do take care of the animals in that way to bring up this information. With this, thank you for the patient hearing and thank you for allowing me to speak here. I hope I have done my justice. Thank you very much. Any question or anything? I don't know if I have made sense, but yes, please.
Nice talk, sir. I just want to know how the REM sleep is associated with the dreams. Is there any connection?
No, connection is absolutely there because most of the dreams, earlier we knew the dreams are coming only during REM sleep, but now we know during non-REM sleep also you might get some dreams. But so far what we know of, you do not remember the dreams of non-REM sleep. So there is no doubt about the connection. And what I propose is, REM sleep is a phenomenon when the dreams come. It overlaps. Why does it come? I have my own hypothesis, which I am not talking about. Now, why the dream? Sometimes, why do we dream that you are seeing that you are talking to somebody? Sometimes you are running. Sometimes you are seeing you are solving a mathematical equation. The question is there, why does that come? Then another question comes with the psychologists or some philosophers that many problems might have been solved during dream. There are many things, like some Nobel Prize winners say that they had solved it in dreams like this. Some great people have said by mothers of great people, the mother has dreamt this or that when the child was born. So what are those things? I do not know. I do not think the science knows about it. But we are in the tip of the iceberg. Unless we know these mechanisms, how do we understand that? But it is true. See, first thing is, dreams are not there unless brain is there. If brain is there, then REM sleep is there, then this is coming. After that, what is coming? So unless we have these basic understanding how to reach there, another difficulty is, we are having this, we need to study this in human brain. Now, who will come forward, who will give one's child or oneself to go and do this testing? That is what sometimes, as I said, as an argumentative manner, we are selfish as a human being. Therefore, we try to do with utmost care, even if when we are using the animals, we are using it for human benefit as well as animal benefit. But yes, there is no doubt that we are getting dream that time. Yes, as I said, my work is on 2 aspects. One is neural mechanism, another is function. So in neural mechanism, my hypothesis is there, whatever we have worked. Why REM sleep, why dreams are coming during REM sleep, that also I had proposed, which I have not talked about. But I have reasons to believe why it comes only that time. But that does not mean it may not come at some other time. Why can it come? It is like if it does a mistake, then if the train moves on the same line, it will reach. If you are taking a Rajdhani train, which should go in some other thing, but because of mistake you have traveled in another line, it will stop in a small station. So there is a mistake, error. That is a psychiatric disorder. But we do not know the mechanism. People like us probably is throwing some light in a small way, whatever we can.
Thank you, sir. Thank you very much.
Yes, please.
But then later on I went on to Germany for my PhD. Just a comment that I remember you from that time.
Oh, so kind of you. Thank you for remembering me. I'm sorry if I cannot recollect.
No, it's okay, sir. I think I was just a student at the time with Dr. Gurcharan for.
Yes, I know her.
A PhD from JNU, probably next to your batch.
No, I mean, she did PhD from Life Science JNU and I did not. I did from AIMS, but I know she was 2 years senior to me because after finishing PhD, she joined lab in AIMS as a postdoc. But of course, we are friends otherwise. That is how I know her. So thank you for your introduction and so kind that you still remember me very. Thank you.
Thank you, sir.
So what are you doing? Are you here in which department? Okay, and your name, please? Shivani. Okay, so if you are still interested, probably we can do so. Huh?
Optogenetics.
So you are doing optogenetics here. I saw somebody's optogenetics. You are doing optogenetics here.
I am doing right now the in silico studies because of the pandemic, because my mentor is in Germany. So the in vitro studies are right now stalled, but we are now again.
So in vitro you are doing optogenetics here?
Not here, sir.
Okay.
My studies are being conducted in Germany.
No, I mean if there is anything that we can do, because I am trying to develop those people who are in neuroscience or otherwise if we can do. So this is the beauty of the university system. And I would like to do, for example, I'll tell you, if I'm not taking too much of time, Professor Verma would know, and many of you may be knowing Sudhir Sopuri. Professor Sudhir Sopuri is a plant science. So one day we were talking in JNU over a cup of tea that he was saying the plants, Arabidopsis or something, doesn't flower unless they see a light. So I said that there must be some communication. And we did. Niti was the student who is now chief scientist in ICGEB. You may be knowing Niti, right? So we did the experiment. She used to bring the plant covered with tin foil and all. And it's so amazing. I did this very simple experiment and we showed how in the plants the faster communication is there, even much faster than even in neural system.
So that's the optogenetic mechanism that.
No, that is not optogenetics. Those days nobody knew about optogenetics, even term was not there.
With Professor Sopuri.
Sorry?
Because I was also in ICGB for 2 years, but I was with Professor Sahal and Professor Chauhan.
Dinkar?
Yes, sir.
Okay. Biophysics.
Yes, actually
We were together in AIMS.
Yes.
Yeah.
So after that I went with the phytochrome research, which Professor Sopuri was also kind of supporting me during the initial time of my.
No, with all due respect, I just wanted to say what I am trying to develop here with any other field, because any cutting edge science goes from the borderline with different expertise coming together. I think we should aim for that. And concept is more important than crore rupees equipment. Equipment certainly is required, but then with a concept, probably many things can be done. And for that, we need interdisciplinarity. And that is what it is that behavioral science, that 3 hours talk this morning and next year is for those people. And I am all for it. And I would like to have lectures by the psychology department for my department and many other things. With that spirit, it's not necessarily you have to come to me, but with anybody to do better science, we need to do that. So if you need anything, we have developed, as I said, the zebrafish behavior recording system, animal recording system, all these have started behavior part in last 4, 5, 6 months in my last 12 months stay here. Thank you very much. Yeah, I would love to hear your talk. Yes. Anything else, please? Any other question? Okay, so should we wind up? Or I am overtaking time already? Ah, very nice. Yes.
And this and their behavior.
Okay.
So I will let you know, send a note.
Sure.
And I have to fix up the date. I fixed the 23rd September. Time I have to fix up. It's not from India, from outside.
Sure, yeah.
Okay, good. Thank you. We'll let you know.
Okay, thank you. Then should I go?
Thank you so much, sir, for your insightful talk. We were enthralled by your presentation. I would now request Dr. Swati Tripathi ma'am to felicitate Dr. B.N. Malik, sir.
गाड़ी कहाँ है उसको रखवा देंगे गाड़ी तो ड्राइवर वहाँ लेते पीछे रखे हुए ले लेंगे हाँ डिपार्टमेंट पे कोई भेज देंगे हाँ सर J1 हाँ देन because वो अभी कहीं नीचे रखते हैं
Thank you, ma'am. We would now like to call upon our next speaker for this session, Dr. Maitri Pradhan, scientist, Karlsruhe Institute of Technology, Germany. She was a postdoctoral fellow at Department of Molecular Biology, Max Planck Institute for Chemical Ecology, Jena, Germany. Her PhD thesis was on the title, Understanding the Regulatory Basis of Defense Signaling in Plants. She has received multiple grants and awards like Young Investigator Grant by the Federal Ministry of Education and Research, Max Planck Society Fellowship for PhD, and many more. She has multiple papers published in renowned journals. I would now like to invite Dr. Maitri Pradhan to deliver a talk on small RNA pathways as modulators of ecological interactions. Over to you, ma'am.
So, hello everyone, and thank you so much, first of all, for the nice introduction, and thank you. And I'm first starting to share my PPT with you. Uh, uh, Give me a second. Hmm.
Ma'am, you can share your screen by the option share screen.
Yeah, but then it's asking me the file, but okay.
First, you open your presentation.
Yeah, it's there.
Click on the share screen button in the green color. Are you able to see that?
So
you have to click on the green button. Share
yeah,
screen.
when I click it, it's asking for which one?
First option is screen. Are you able to see that?
No.
What are the options you are seeing?
It's desktop, whiteboard, iPhone.
Ma'am, we can share it from our end as well if you want.
Yeah, that would be great because I have made some minor editorial thing, but it's okay if you could
click on the desktop.
When I click in desktop, it's asking for the system preferences.
You are using MacBook.
Yeah.
So, and we can share our. This student, you can share yours.
It's a problem.
Ma'am, you can just let us know when we need to change the slides.
I think I found it. Give me a... Can you see that?
No, ma'am, not yet. Yes, ma'am, it's visible.
Yeah. So, sorry for the delay. Good afternoon, everyone. And again, thank you so much to both of you for a nice introduction. And secondly, thank you so much, Professor Verma, for the invitation. I'm really grateful to talk to you all guys about my research, what I'm doing, what I have done, and what's my future plan. And I hope you had a great lunch, food, which I'm missing here. And yeah. Moving forward, so the topic of my research is small RNA pathways as modulator of ecological interactions. Going for that, I think it's not, the slides are not, so it's working like a PDF. It's not like PowerPoint presentation. So little bit problematic. But yeah, as we all know that plants do not grow alone in nature. They have partners. They have interactomes. The interactomes starting from herbivores to pathogens to mycorrhiza fungi and other microbes. And how this function? It's like when there is a biotic interactor, and they interact with the plants. There is a cell membrane, and then the cell membrane we have different type of receptors, and these receptors get activated and disactivate the MAP kinase and calcium signaling, and that activates the master transcription factor. And these master transcription factor like MIX and MIBS and all, they activate the phytohormone signaling, and which then activate the MIB signalings. And at the end, that result in the secondary metabolites for defense. Okay, and this is the traditional protocol or the method that people know that this in this pathway it acts. But our question was whether small RNAs has a role in this pathway or not. So before jumping into small RNAs and confusing all of you on that, I will give a brief description about what is small RNAs. So small RNAs are non-coding RNA molecules which are 18 to 30 nucleotide long. They identify their targets. Can you see my cursor? Yeah. Yeah, okay. They identify their targets through complementary base pairing by forming a RIS complex here, RNA-induced silencing complexes. And there could be two kinds of small RNAs. In plant, I'm talking about, which are majorly studies, which are microRNA and siRNAs, small interfering RNAs, and they have their different type of biogenesis pathway. So miRNAs are produced from miRNA gene through pre-miRNA pathway. They form a loop-like structure, which are then cleaved by dicer-like protein, forming a double-stranded structure, and then the functional miRNA get loaded into the RIS complex. And then the transcriptional degradation or translation inhibition happen. And then the siRNA pathway. The siRNA could be formed by different sources, and which could be formed by RNA-directed RNA polymerase and followed by and clipped by dicer-like proteins, and then forming a single-stranded siRNA, which is again loaded on the RIS complex. Basically the RDRs and the DCLs are the most studied proteins in the small RNA pathway. And here the Argonauts protein act as the effector protein for the transcriptional degradation or translational inhibition. Again, coming more onto the Argonaut proteins in the small RNA pathway, these single-stranded RNA, when they are loaded in the Argonaut protein, this Argonaut protein with the small RNA duplex, they scan the target. So they scan the target and forming the RIS complex, and which giving rise to the translational inhibition. And in the plant, we have found that it's a large family of protein, and they have large number of genes. So traditionally, people think that it has only function in development, differentiation, viral infection, and it's a common belief that it has a redundant function. So we ask ourselves that, is it really a redundant? So Argonaut has a redundant function or not? So when I started my scientific career in India, my previous colleague and my supervisor in his lab, they found out that this Argonaut protein go through a duplication event. Basically, in a multicellular organisms, they go through 5 duplication event, which give rise to this big family of a gene, and from a unicellular green algae giving rise to several genes, and they divide this into 4 different classes, or we called it as clades. And these evolutionary changes that, or the duplication event happen in the Argonaut, that change in the sequence and the domain structure of the Argonauts, which give rise to the different gene function. And they also find out that this dynamic evolution of agos have produced the signature of functional diversification in the small RNA pathway in higher plants. So then having this knowledge, we thought to translate it. And the translation, we wanted to do it in a plant species that is called Nicotiana attenuata. So Nicotiana attenuata is a model plant, and it's grow in a post-fire environment, and it's a desert plant, and it's grow in southwest of United States. So I started my PhD here in Max Planck, and the first time I saw this plant, and it has so much of nicotine, you can barely stand in the greenhouse for 1 hour and feel dizzy. So no doubt that why the ancient Native Americans use this as a weed. So yeah, so this grow as a monoculture population in varying density. and it's attacked by diverse communities of microbes, and they coexist in the field with all these mycorrhiza, with the pathogen, and with the herbivores. So that is very exciting for me, that in the field condition you have so much of interactome, and how you make yourself so resistant to it and having the highest amount of nicotine in your body. So and it has the multiple induced chemical defenses, and it has been studied since last 20 years. So when this plant is attacked by herbivore, it produces multiple secondary metabolites, starting from nicotine to diterpene. It produces trypsin, which is modulated by phytohormones. There are different type of phytohormones. The important one, jasmonic acid, salicylic acid, ethylene, and abscisic acid. And these phytohormones are regulated by different transcription factors, working mix the major ones that we all know. But by the time I have started my study, nobody knows that what these agos are doing in Nicotiana, because in Nicotiana, people already in my lab already studied about RDRs and DCLs. It's a complete functional characterization already done. But what is happening with the argonauts, whether they're involved or not, that was the big question. So I took it as a challenge, and having all those knowledge from my previous colleague to stand on their shoulder and to giving their respect, I took the information from them, and having collaboration with my bioinformaticians and all, we found out that these argonauts in Nicotiana attenuata have 8 unique conserved genes, which are argonaut 1, 2, 4, 5, 7, 8, 9, and 10. And AGO1 has 3 splice variants, AGO4 has 2 splice variants. And when I started it, I found out that what should be the process, because as I already told you, this plant is in the field is attacked by different kind of interactomes. So my first hypothesis, how we are going to answer the functional characterization of these argonauts. So the objective was to explore the biological function. And how we wanted to do? We wanted to take 3 interactome. First, of course, will be the herbivory, because this is what the heart of our study, and then followed by pathogens and mycorrhiza. These are the 3 challenges I wanted to take, and to uncover the functional specificity of AGO function, because it's not that I have all the 8 agos and they can be function in one interactome. This cannot happen. So what is happening on there? And also, because I am working on the ago effectors, the end thing I wanted, that how these small RNAs or the microRNAs are regulating, but in an ecological context. So the approaches that I followed was the loss of function characterization, followed by their biological function test in the nature, it means in the field, also in the greenhouse, and molecular evaluation of signaling and defenses, and followed by the microRNome, how these microRNome are programming in a temporal way, or what is happening there, and identifying their microRNA targets, validating the microRNA function, and then integrating this AGO function along with the other components like RDRs and DCL, so that I can have that pathway fully, I can show that how the pathway is functioning. So what I did was I did a time course experiment. What do you mean by time course? It's a kinetic experiment. For the biological function, how to explore the biological function, I took a wild type plants, and then I did the wounding, and then put the oral secretion of Manduca sexta. So Manduca sexta is the caterpillar for this Nicotiana attenuata plant in the field. So we nurture them, we grow them in the greenhouse, and we collect the oral secretion, and we do the time course experiment. So in the wild type plant, what I found when I did the wounding and oral secretion experiment, only Argonaut 5 and Argonaut 8 are elicited, and they are significantly different from the control plants and water treatment plants. None of the other Argonauts, they show any differences in their elicitation pattern. So the first click was, okay, it's only 2 Argonauts which are showing this kind of expression pattern. Then we wanted to see that, okay, we have a toolbox, so why not to go for that? So in the toolbox, we did the loss of function characterization, and we have the knockdown lines individually. So we introduce the knockdown lines to the herbivores, and we found that only Argonaut 8 lines showed a significant increase in biomass of these caterpillars. And Argonaut 1 plants, when we got these knockout lines, we did not get a completely transformed plants, because they are in traditionally, it's also already reported that Argonaut 1 is involved in the development and growth. So we could not get Argonaut 1, except that we could not see any of the differences in the other lines. So in Manduca sexta, caterpillar grow better in Argonaut plant, which validate that the Argonaut 8 plants, are involved in the herbivory. I'm not showing here the Argonaut 5 because this is in process, and we also found that Argonaut 5 plants are also compromised with the fitness here. Then we thought that, okay, the plants are compromised, and which are the defense compounds then that are compromised because of Argonaut 8 silencing. So then we checked the anti-herbivore defenses compounds. We found out the major important nicotine is down in both the lines of Argonauts. The DTGs are down. We also checked the important genes of the nicotine production and the DTG pathway. They are significantly down in Argonaut 8 lines. We checked the trypsin protease inhibitor. They are down in Argonaut 8. Check the genes regulated for the propanoid and the phenylamide pathway, the PAL1 and DH29, they are down regulated. So, and also we check the MIB8. So the major transcription factor is also down regulated. So we concluded here that these major transcription factor regulate the genes in the pathway, which result in the compromise of anti-herbivore metabolites. And then we found out that, okay, this AGO8 is there in the RISC complex that attached to the microRNA and then regulating the defense function. Then we wanted to know what are the miRNA accumulations are happening, which are AGO8 dependent modulation pattern. So we did the microRNA analysis, and in the heat map, you can see the green one is in wild type, and you can see there is a clear difference in wild type pattern and in AGO8 pattern. So the green one is, you can see in wild type, there are microRNAs which are expressed only in wild type, and they are disappearing in AGO8 line. But there is also a group of microRNAs which are having temporal dynamics. It means they are only expressing in certain time point during the herbivory defense. And we did it in a kinetic experiment where we found out that the herbivory induces change in the accumulation of microRNAs in a time-dependent manner. And due to silencing of these AGO8, there is a complex pattern of the differential accumulation of microRNA that are happening from 0 time point to 18-hour post elicitation. And we found that AGO8 is important for the modulation of miRNA accumulation during herbivory. And then we determined how these microRNAs are related to genes which are relevant to the compromise of defenses after herbivory attack. But we found out there is a 1-to-1 and 1-to-many relation. It means there is a MIB gene, which is, we found that there are 3 microRNAs which are targeting MIB genes. We also found this particular microRNA is also targeting Werke and MIX. And this 6-to-8-1 microRNA also targeting DH29 that lead to compromise of phenyl polyamines. In this in the other case, like nicotine, we have found only one microRNA that target the nicotine producing metabolites gene pathway. So in this case, what I said already, that we found there is a many to one and one to many relationship, and we inferred that AGO8 dependent microRNA form a additional layer here for the regulation during herbivory. So that was the microRNA analysis we did. So after, so I will conclude this finding as that we found 11 Argonauts in Nicotiana attenuata, and only 8 unique Argonauts are conserved genes. And these 8 genes, in these 8 genes, only Argonaut 8 and 5 are specifically elicited during herbivory. And we also had the knockout lines, and but we could not found that any other Argonauts could be involved. And also, we found out that there is in Nicotiana attenuata, there is a microRNA which is reprogrammed in time-dependent manner. And also, we integrate our result with the other components of the pathway like RDR, DCL3, and DCL4, which are already characterized in this system. Then we ask that then what is happening to the other Argonauts, and we found out, and we thought that okay, let's give the let's do the biofunctional characterization of other Argonauts for the pathogen and for the mycorrhization or the symbiosis. So followed by this story, I did a small experiment in the greenhouse. So I did an infection assay on wild type plant. I elicit the wild type Nicotiana plant with Fusarium spores, and I found out that only Argonaut 4 is elicited after Fusarium infection. And also I validate that with the lesion diameter, which is significantly higher in Argonaut 4 line, and there is not the other Argonaut lines. So then we thought, okay, we have a very interesting result for the pathogenesis, and we have a great opportunity because we have the field in our hand in Max Planck. So let's take all our knockout lines and grow them in the field. So I did a 2-year field study, being there in the field in United States, Utah, for 2 years, 4 to 5 months. Then we grow the plants in the field. And I checked the phenotype of the plant. So these are like 700, 800 plants together of all the Argonauts line. So you can see in the field, the Argonaut 4 lines, these are the wild type lines, which are asymptomatic, and these are the infected Argonaut 4 plants. After 12 days, they start to wilt, and after 16 to 18 days, they die. So we have a very clear phenotype of the pathogen infection in the field. And the good thing is that before going to the field, my other colleagues already identified the Fusarium species that I was using from the field only. So we know that the Fusarium that we are using, it's there in the field. So you can clearly see after 13 dpi, the plants, the lesion diameter, the necrosis, the chlorosis, the wilting is very high as compared to wild type. And on 16 dpi also this pattern follows. So then I took all my plants to lab from field, and the question was why and how this is happening. Because we are working on a mechanistic mechanism way. What is the mechanism that is following? So we did a hormone profiling of the Argonaut 4 line and wild type plant, and we found that jasmonic acid is the phytohormone which is compromised. To confirm this, we did the complementation assay where we complement jasmonic acid and methyl jasmonate to the infected plant, and we could clearly complement and restore the phenotype as the level of wild type. And you can see in the mock treatment, it's significantly high. And after jasmonic acid and methyl jasmonate treatment, they are in the same level in the lesion diameter. We checked also the gene expression study in the jasmonic acid pathway, and the key genes like OPR3 is compromised. We also did the small RNA profiling here. We checked the target study. We did also we also wanted to check which are the RDRs and DCLs that are also involved during the pathogenesis, and we found that RDR1 is the one which is involved here. And also, as I already said, that we validate this in the field, and we can clearly see that the Argonaut 4 lines are more or highly susceptible in the field condition. And we found that Argonaut 1, RDR1, RDR2, and DCL3 are also susceptible for this fusarium species in the field condition. So this is what we found. So to conclude this pathogen story, I would say that from these 8 unique Argonauts, only Argonaut 4 is involved in the pathogens registrant. And along with the Argonaut 4, RDR1, RDR2, and DCL3 are also involved in this process. They produce the miRNA, and we also found that which are the miRNA and which are the 24 nucleotide siRNAs these that lead to the resistance for AGO4 or for the pathogen resistant, which is through the jasmonic acid pathway. And we found that the role of AGO4 dependent small RNA pathway in plant resistance against pathogen was validated also in the field condition, because that was our motto. Because there are many phenotypes that we found in greenhouse we could not replicate in the field. And the beauty of the field is that there are many phenotypes we could not found or we hypothesize in the greenhouse, but or we could not see any phenotype in the greenhouse, but as soon as you put them in the field, you saw the phenotype there. So that is the beauty of the field, and that was a very important aim of ours, that to validate in the field condition. So this was the story of the pathogenesis. Then we thought that, okay, we have 2 Argonauts which are herbivory specific. There is 1 Argonaut which is pathogen. So is there any Argonaut involved in the mycorrhization or the symbiosis? So to do that, I did an experiment with the competitive fitness, where we grow wild type plant and Argonaut plants in the same pot with and without mycorrhiza. And we did it for all the Argonauts except Argonaut 4, 5, and 8, because we already characterized them and published them. So we checked Argo 1, 2, 4, 10, and 7. And we could see after growing them with and without mycorrhiza, we found only Argonaut 7 has a fitness phenotype. So with respect to stalk length, and also with respect to seed capsule. So when we use silence Argonaut 7, they have shorter stalk length and lesser capsule. And we also found out that the 11-carboxyblumenol C is higher in Argonaut 7. We checked the Abascals number, vesicles, and hyphae. Interestingly, they are higher in Argonaut 7. So the first impression was, okay, in a resource limited condition, Argonaut 7 has compromised with fitness, but with higher AMF colonization. So we conclude AGO7 specifically involved in competitive fitness and AMF colonization. To follow this, what we did a microRNA profile. We found many microRNAs which are only modulate during symbiosis in AGO7 background. Among them, I am giving 2 examples here. One is the conserved microRNA 473, and one is the novel Nicotiana specific microRNA. You can see there is a early and a late elicitation of these microRNA during symbiosis. And we also check the target genes of this microRNA which are compromised. And these target genes are specifically DELA genes and ethylene specific pathway gene insensitive genes. And what we did So we have this microRNA. As I already said, in Max Planck, we have a very nice toolbox. So using that toolbox, I overexpress this microRNA, and I checked the target genes, what we previously got from the microRNA analysis. And I found out that, for example, I'm saying here, the microRNA 473 specifically targets to fatty acids, lipid metabolism, and DELA pathway. And in the overexpression line, these are all compromised. And we also checked the colonization event and the capsule number also. So this is the phenotypic characterization we did with the overexpression line. So in the Blumenol C here, you can see after overexpressing the microRNA, the colonization increases and the capsule number decreases. So as summarizing this story, I would say that Argonaut 7 is involved in modulating the mycorrhiza colonization in competitive fitness in nature, and AMF colonization reprogram the miRNA accumulation, which is AGO7 dependent, because we see a very clear phenotype when we silence Argonaut 7 and we do the microRNA profile. And there is a very clear signature of those microRNAs. And these microRNA target genes are the key signaling genes in the phosphate starvation, fatty acid, gibberellic acid signaling, ethylene signaling, and also auxin signaling. And that is important for the competitive fitness. To summarize all my three stories, small RNAs are including microRNAs, which form an essential layer in the regulation of host that processes the direct, the deleterious and symbiotic outcomes. And what we found that in the insect interaction, there is Argonaut 8, which is involved, and as an important component as an effector that control the herbivore interaction, and we could validate the hypothesis regarding the functional of evolution also of the argonauts. And Nicotiana argonauts modulate the host adaptive responses and possesses a functional specificity. Why I said a functional specificity? Because when you change the interactome, for example, to a fusarium or a symbiotic fungi, then there is specific microRNAs which get loaded in the RISC complex. So that's why I called this functional specificity of argonauts is entailed by recruiting unique small RNA specific in the nature of attack. So if you have insect, then the specific microRNA will be different than with fusarium or mycorrhiza. And these are deployed to modulate signaling and response towards their changing in the biotic environment, and which help the host to mount the appropriate response and to discriminate who is my friend and who is my enemy. So having this knowledge, from my PhD and from postdoc, after coming to Karlsruhe, now what I am doing is how these plants simultaneously engage with friends and foes under resource limited condition. Because the plants never grow alone in the nature. They always grow with friends or foes or pathogen or symbiotic organism. And while growing in the same environmental condition with a resource limiting condition, how this small RNA move from cross kingdom, like from pathogen to the plant or plant to pathogen or symbiotic fungi to plant, how this cross kingdom function of small RNA act as a communicator or a signaling molecule or a defense molecule in this triotrophic, or I could say that in a multitrophic interactome, because traditionally people only talk about host interaction with host or a pathogen or host or a symbiont. But this is what I'm putting forward, that in a multitrophic interaction, how a host can discriminate who is a friend or who is a foe. So this is what my future direction of my study. And At the end, I want to thank to my previous supervisors, my mentor, Dr. Shuprakash Pandey, Professor Baldwin, and now Professor Regina for being there always and motivating and helping me out and criticizing also the good criticism, which always drive you to work more and work on yourself. The funding agency, the BMBF Foundation, the Ministry of Science for funding me, for providing me bread and butter, I would say here, and Max Planck Society for funding my PhD, and my group member, my past group member also, on whose information I stood on their shoulder and move forward in the project. And now my current group member. Thank you so much, and thank you so much to all of you for your kind patience, and thank you for your attention.
Thank you very much, ma'am, for your insightful talk.
Thank you.
Now the audience might ask any questions if you have.
Hello, ma'am.
Hello, sir.
It was a nice talk. Indeed, a great work you have done. So actually, I'm more concerned about these microRNAs, and
yeah,
it was
sure.
exciting for me and always confusing also. Because, I'm confused whether these microRNAs, they are specific to particular gene or gene families, or one microRNA can act against many of these gene families together.
Yes, as okay.
That's my question.
Yeah. So what we found that a single microRNA, first of all, the microRNA are specific to interaction. So there are specific, for example, I'm telling 473 that I'm working on. This paper is now under revision and we are going to submit soon, I think in 2 days, that 473. This 473, I could not found it in herbivore interaction or pathogen interaction. I only found this microRNA in symbiosis. But I found it, but it's not significantly different. The criteria that we choose to filter it out, this doesn't come there. And also what I showed that this microRNA 473 can target 3 pathways. Okay, one DELA, the gibberellic acid pathway, one fatty acid pathway, one lipid pathway. Okay, so it has 3 targets pathway. As you have this question that it can target to multiple, yes, it can target to multiple pathway. Going back to
the mechanism is same for, I mean, when targeting multiple genes or multiple gene families, the mechanism.
Mechanism in the sense, what do you mean by mechanism? Like the way the process
act against multiple gene families. That's what I'm asking.
The mechanism in the sense how they identify the gene, right? Yeah. Because the microRNA will attach. So in the RISC complex, as I told, the AGO is the effector and it will attach to the microRNA. Yes, because the Argonaut has the 3 domain, PV domain, mid domain, pass domain. So it has 2 sites, one hydroxyl site and one 5' end site. So it will detect only the seed sequence, which is 6 to 8 nucleotide. And yes, they bound to that only. Yes, I would say yes, the mechanism is same. What we informatically know. For 473 immunoprecipitation, I have not done that. So I cannot specifically tell you that, yes, in the immunoprecipitation assay, we found, yes, it bound to exactly the same position. But informatically, in a bioinformatic analysis, yes, we found that, yeah, the mechanism is same. Yeah. And for the second question you asked, that do they also like microRNA target to one gene? Yes, as I said, in going back to the herbivory study, there is the PMT gene, which is like we found only one microRNA that target to the PMT gene that compromise the nicotine pathway. So yes, there could be that one gene, one-to-one relation or one-to-many relation. Yeah.
Thank you. Thank you very much.
Excellent presentation.
Thank you so much, sir.
Congratulate you.
So much.
Now, in nature,
Yeah.
mixed population of microbes.
Yes, sir.
Fungi are there, pathogen fungi are there, fusarium, and then caterpillar. And how about nematodes? How they take care of nematode at the same time?
Thank you so much, sir, for this wonderful question. During my PhD, because we were more focused on herbivore and the fusarium and rhizophagus, I could not get a chance to work on the nematode.
Okay.
But yes, if we go to previous study, it's already found in root nematodes that Argonaut 7 is involved in root symbiosis in the nematodes, which is involved in the nodulation. This is already identified in nodulation. But in my case, I have not checked any Argonauts in Nicotiana specifically that is involved during nodulation. But it would be very interesting and very, very interesting from a personal side, working on a very big gene family and all, that to see if there is any Argonauts that has a role during nematode infection. Yeah.
Good. You have done very good work at Vienna. I was in Vienna many times.
Yes.
I was there 3-4 years earlier.
Mm.
So kindly convey my best regards to Natalia.
Sure, sir. Sure.
Thank you. Thank you very much.
Thank you so much.
All the
Thank
best. All the best.
Thank you so much, sir.
Thank you very much, ma'am, for clearing all the queries. Now, the final speaker for today's session is Dr. Shivani Sharda, ma'am, from Amity Institute of Biotechnology, Amity University, Uttar Pradesh. She has her PhD from Max Planck Institute of Bioorganic Chemistry with affiliation to Heinrich Heine University, Dusseldorf, Germany. After that, she did her postdoctoral research from International Center for Genetic Engineering and Biotechnology, New Delhi. Her research expertise is in genomics and proteomics, and she has published multiple research papers in various journals. Now, I would like to invite Dr. Shivani Sharda ma'am to present her talk on application of optical modules in optogenetic and optoproteomic genome modulation. Over to you, ma'am.
Good evening, everyone. Good evening, Verma sir and Malik sir. And thanks for giving me this opportunity to talk on a thematic view of the work that I have performed again during my PhD program and later on in my research interests that I have pursued here in ICGB, followed by Amity University. So my work was basically on bacterial phytochromes, cyanobacterial phytochromes, and these are the signaling proteins, which are photoreceptor proteins. And I worked on doing crystal analysis, kinetic analysis, and many site-directed mutagenesis studies to begin with. And we are right now doing some analysis along with my mentor, who is Professor Wolfgang Gattner. He's an emeritus professor in Leipzig University. And we are forwarding our work into terms of optogenetic modules. This is a long set dream. Let's hope that we are able to conclude some information in our current studies. So I will just have a brief introduction of how aptogenetics started. And as I said to Malik sir, that it was the first state-of-the-art experiment which was done on a neuronal analysis with a neurotransmission. So it is an integration of optics and genetics that allows us for the experimental control of events at both cellular and at molecular level, even at the subcellular level with a specific signal intensity. So the basic process that goes here is expressing a light-activated ion channel, usually isolated from bacteria, in a specific subpopulation of neurons. That was the first state-of-the-art experiment, happened in 2002, followed by Carl Deseroth, who has given a nature method of optogenetics in 2010, which he has proved that using bacterial opsins, he could regenerate or he could restore some of the lost sites in rat neuronal cells. So that is the first level of optogenetics as a conceptual form. Now, what we have here in optogenetics is that it has a very high spatial and temporal control over the processes that we want to control. And that is where the entire photoreceptor domains was a talk of the town. And because we have a spectrum that is from UV to the visible and infrared range, there were many people who were working in the different forms of the photoreceptor unit. So the population of the photoreceptors were being accumulated and worked about for their application part in various fields, beginning from neurosciences, because it's the most connected situation with the synaptic connection here. So the population of neurons that were being targeted in this was modulated, and the nature of the modulation was all under the experimental control. A very good system, and then it has been proven at a cardiac level, which means at cardiac muscle sense, and now at all the disease models. My work further here was actually on reactive arthritis, a type of arthritis which is because of infection. And the infection is very closely related to inflammatory bowel disease. So it is exactly not really connected to my photoreceptor research, but I made a very strange connection to make utilization of these photoreceptors as photosensory modules to see if I could see the perturbations of the infection-related cells at the level of the biomolecular studies. So a very diverse toolkit is available with us, either as single compartment, as dimeric proteins, as multimeric proteins, or as chimeric proteins. Now, most of my work was on red and far red infrared proteins. These were later on coined by Roger Seen, a posthumous. Right now, he is not with us. Roger Seen as near infrared spectrum. So that is the terminology that was given, and we were given a new terminology, which is infrared fluorescent proteins. It has a very high intensity tissue penetration, and the good point about this is that it is not having any kind of what we say as side effects while it is penetrating any live tissues. So we have a repertoire of different photoreceptors. I am just going to give some different details of what we have performed in our studies and how we are going to proceed, though there are some which is going into a paper, so we are not disclosing all the kinds of inserts and the clones that we are preparing, but in silico studies are being already told here. So now the optogenetic effects on any cell would depend upon many factors. The factors are majorly what you are handling. Either it is single component, it is multi-component, you are mixing two photoreceptors or controlling through two different light signals. Efficiency of the expression in any cell format. Now it could be, we began with E. coli, we have moved to yeast, and we are probably moving very soon to human cell lines or ES cell lines here at Amity itself. So the source, wavelength, as well as intensity of the light and the location of the target cellular and membrane interface being investigated. So this is just a very brief overview of a paper, which is the most recent one, which is giving you the spectrum of light signal photoreceptor proteins or modules that are available with us. So we begin with UV range and going up to the infrared range. Most of my work has been the range of red and infrared range here, but in my lab, because it was a lab that is related to photoreceptor protein analysis, we also worked very closely with Italy, and we have worked with LOV, which are light oxygenate voltage gated channels. So there are different versions of how you could perform the work. So when we talk about the UV or the blue light receptors, we have the LOV, CRY receptors, which are mostly utilized, have been the most common utilization after the first proof of concept, because they have a very good allosteric control by having the connection with FMN and FAD, and then we could use them for doing the modulation at the cellular interface as well as the molecular interface. So the entire process of control depends upon how we're going to proceed with the protein intensity, their purification, as well as their stability. We call these proteins as bi-switched proteins. It's a switchable concept. Now, what does it switch to? It switches to both active and inactive form. The active and inactive form can run from seconds and go up to minutes to hours. It all would depend upon how and where the control is lying for this analysis. So we have a repertoire of allosteric control, either oligomerization and dissociation, heteromerization, and self-cleavage. 4CL is the latest technology where they have utilized it for a protease action along with the 5B phytochrome proteins. Some of the optogenetic engineering domains with the light-gated channels are the light-triggered nucleoplasmic translocation, where most of the genes with the optocrack model have been used to see that they could modulate the gene expression of the target genes. Similarly, we have photoactivable CRISPR-Cas related genes with mechanisms that are again controlling. All of them are really falling under the UV range. When we say the photoviable activated base editing mechanism, we have specific bases in the protein sequence which have been identified through site-directed analysis as to which are the controlling units in making the light intensity work. So here I am more interested towards the Cree-FLP system, the Cree-flip system, as well as the photo domains of red far red light for now, and very soon we are going to have a chimera model with the LOV domains. Now, just to give a bit more detailed analysis, this is the range of our spectrum. I have worked on phytochromes basically with various domains which are part of this protein. These domains have chromophores which bind to these proteins. So we have two different canonical units in the cyanobacteria that I worked with. One is biliverdin and the second one is phycocyanobacterin. They both have two different proteins in the same cyanobacteria, which was utilized for many different kinds of kinetic as well as structural studies. Phototropins, as I said, was closely worked in with our close collaborators, LOV domain proteins with Italy. And we had cryptochromes being worked in with a Spanish collaborator, which was worked mostly in plant work. So in this process of getting or setting my goal, I have worked on CBCRs, which are related to calothrix, with two different proteins which were present in the genome of calothrix 7601. The strain was kindly provided by Tandio de Mersec from Paris. It had two different parts of the phytochrome. One was a PCB-linked protein. We renamed this as CPH because it was cyanobacterial phytochrome A-type, and there was a biliverdin-linked protein, which is PCB or CPHB, because it was linked or having a different chromophore. The usual spectrum, what you see here on the left, is actually generating you the version of the protein. The total protein and the content with purified forms, and this is the active to inactive form of the proteins that is present here. As you can see that the oscillation of the protein and the ratio that we could generate was 1, which means we could actually make a complete switch on and off in a matter of 3 to 10 seconds. These were the kinetic studies that has been performed on the entire protein and part of the domain protein. So what were the domains that were present? We have the per-sim domain, GAF domain, phi, histidine, kinase domain, which was modulating itself to a response regulator protein. Now this process, we changed our version of taking the entire protein, because if we talk of the entire protein, this is a 90 kilodalton protein, which was a bit more difficult to really handle while doing purification studies. So what we had decided is that we connect ourselves to the shortest possible domain of the protein, which is still photoactive and provides us with the more viable, I would say, switch that we want to use. So what we did was that I had taken out some of the domains or truncated my protein into different domains. We took basically the PAS GAF phi domains because they are the major areas of the protein where the chromophores are supposed to bind. Then we had only the PAS and GAF domain, only the GAF phi and kinase domain, and kinase domain only separated out. So this process was done by doing various cloning efforts, and this is what is being still continued, because we are zeroing in down with the kind of site-directed mutagens that we have to see that we could have the least possible score of a protein that is viable for us to work with. So to give you the brief idea, we can see that this is the chromophore that is bound to the protein. And if we do any site-directed mutagenesis here, we are supposed, or we would rather have no interaction with the chromophore. The light-activated switch is going to go blank. This process or this understanding has come from various studies. We had also done some kind of simulation patterns for these studies. So we have the knotted or knotless cytobacterial phytochromes in calothrix here with us. Some of the studies with the domains that have been done is the kind of purification profiles that we have performed. So we have the PAS GAF domain, the CBD domain, which is the beginning of the protein till the binding domain, the PAS GAF phi domain, only histidine kinase domain. The B and the A here denotates actually the B and the A type of proteins that we have. Now, out of this, we have more interest towards CPHB protein because it is having a cognate chromophore binding with biliverdin. Biliverdin is present in mammalian systems, so we are more interested in actually using that system for analysis. That is why we had done some expression studies, or we can say the overexpression studies in various types of cells of E. coli, and some similar studies are already being conducted for the eukaryotic backdrop. So this is some of the studies after the domain interaction was completed. So we have PAS GAF phi domain, we have only the PAS and GAF domain. And if you just clearly see parallelly, when we have removed the phi part of the domain, we lose most of our controllable switch. We only are left with a very less amount of the protein, so the amount of protein is not really co-regulated, or it's not being able to be overexpressed in spite of the best of efforts that we could perform. In a PAS-GAF domain from CPHB, a shorter version of the entire protein, we lost the entire function of the switch. So why these studies are important is because we can have tunable properties of this protein as and what we are requiring our work to be performed. Some of the structure function studies that are being carried out are the crystalline analysis, though we are really struggling with the full form. So we are concentrating our work towards the GAF-phi domain, which has the highest binding towards the chromophores. Now, this is the base of a photoreceptor studies. How are we going to utilize in today's time? Because I had very diverse kind of research analysis. I had worked on systems concept on reactive arthritis, where we had done metabolic reconstructions of the infectious bacteria that are inflicting humans. And from there, we have found some of the genes which are actually correlated amongst all the microbes. So it's a microbe, and a host microbe interaction that we have followed. And we have found out some target 4 genes, and out of this, 1 target gene is common amongst all the 7 different microbes which are infected the human systems in reactive arthritis and IBD commonly. And we have found, we have used this as our target analysis to see the inflammatory changes that happen at the level of cells. So as a proof of concept, we have begun with one of the type of cells, which are TH cells, to see whether there is a change depending upon the modulation that we provide with this target gene. So some of the further studies of bacteriophytochromes, where we have tried to target many different places where the site-directed mutagenesis could be conducted, and we have yielded some of very different kinds of results, especially with the cysteine, leucine, valine, and methionine at some positions. The changes, we have very unique what we say as site-directed mutants, which would be utilized later on as our photo modules. Some of the in silico studies that we have performed as the new proof of concept here is, this is our gene or our protein, which is the entire protein. And this has been having a docking analysis and a fusion protein analysis with an inflammasome. Inflammasome has many different genes, and they are responsible for the macrophage switch or the inflammatory switch. And because, as I said, I worked on inflammatory bowel disease and autoimmune disorders, this is where I connected the work together. We have done all the kind of studies which are in silico studies to perform the analysis, where we have found the exact residues where this is the inflammasome target, one of the gene, which is caspase, and we have CPHB structure with the PAS and the histidine kinase domain residues being interacting with this domain. So we'll be performing our next studies in our switch with this kind of a genetic circuit, and we will be using PAS-GAF, PAS-HK, and GAF5 domains as our modules for beginning our optogenetic studies. So basically, it is a design concept which will go from genome or gene editing towards synthetic systems or synthetic biology. The synthetic biology systems that we have is that we had already done the computational analysis. Now we are at the interface analysis, where we are performing or in process of standardization of the in vitro studies related to the inflammatory cells with infection and without infection. The initial studies, we are still standardizing the protocols as of now, but the process is that we have already done the in silico analysis. We have found some of the controller units as part of our optogenetic modules, and we are going towards understanding the outputs in various methods. So this is a brief version of the optogenetic systems. Again, this is a unique concept. For now, the concept is under review. The paper is also under review with the preliminary studies being conducted with our coordinators, as I said, in Germany, and in silico studies majorly happening here in India. So the significant resolution that we have got is, this is the very first time that we are using inflammatory models or auto-inflammatory models to find out if there are changes at the level of the cell interface system. For this, I have also another collaboration with a nanobiotechnology expert. And there is a project that we have almost completed with nanoalginate gels. So we have combined the third concept. Using this, I couldn't really go into all the details here because that is, again, the second paper is under review, where we have used the nanoalginate gels for the structural elucidation. Once we have completed that, we'll be putting our protein, which is our genetic module that you have just shown, the PAS-GAF5, the GAF5 or the GAF-HK domains, onto these nanoalginate gels along with our target genes, and they are going to record the the chloride as well as the sodium efflux channels, the potassium efflux channels at the interface of these cells. So this concept is what is being reviewed and worked upon. It's completely new. We are still kind of finding out the ways that how we are going to stabilize our proteins or localize our proteins on the nano-alginate gels. This is the work that is happening again with one of our collaborator, Dr. Rachna. So we have three different parts. We have experimental part, which is completely proteomics. We have an in silico part or systems biology part, which has happened on a disease or let's say complex disorder. And we have nanogel structural elucidation part, which is based on doing the nano analysis because they could become the sensors or the actual devices at a later unit. So we are combining three of these expertise together. There are four of us which are working in this direction under the mentorship of my guide, as I said, from Germany. And we will be just hoping that in this coming year, we'll be generating a data that is eventually available for the experimental analysis. So that's a brief talk that I had for the application of these optogenetic and optoproteomic approaches in the disease models that we are trying to find out. Thank you very much. We are open for any questions here. Thank you, sir.
Shivani, just curiosity. Why did you go for complex calothrix? Why not simple FNO-CAPSA, FNO-TC, like that?
Sir, actually, calothrix cyanochromes or cyanobacteriophytochromes, we considered them as an evolutionary switch for special proteins, especially related to the photosystems. We have some of these cyano proteins which are present in only few of these evolutionary switches, and they have all the modules, all the types of proteins present in their genome. So the main focus nowadays have shifted from the blue light receptor to the red and infrared, because red and infrared has, as I said, a very high amount of tissue penetration and very less side effects as of now has been studied for this. So the spatiotemporal control is very good. We have seen it is better than the LOV proteins, but we are still trying that we could make a chimera of both LOV and cyanobacteria have both LOV as well as phytochromes in their genome housed in 4 of the cyanobacteria. That is why I would rather prefer cyanobacteria than any other bacterial phytochromes.
Okay. Thank you very much.
Thank you, sir.
Thank you so much, ma'am, for your wonderful presentation. Now, I would like to call upon Dr. Surbhi Dabral, ma'am, to felicitate Dr. Shivani Sharda, ma'am.
Back to you because I have a work which is also interesting for the neuronal culture that I had done before. So because I was the person who established the lab at Kunalakdev University, so I was really interested. If later on this process could be happening at the synapse, it would be much easier to see as compared to what we are trying to do for now. Can I take leave?
Thank you, ma'am. We do not need magic to change the world. We carry all the power we need inside ourselves already. We have the power to imagine better. With this thought in mind, I would now request Dr. Shalini Porwal, ma'am, Assistant Professor, Amity Institute of Microbial Technology, Amity University, to please come up on the stage for the concluding remarks.
Thank you, Pavitran, and thank you, Ishika. I'm extremely thankful to all the esteemed dignitaries and intellectuals who were part of this 2 days TBT-sponsored international conference on emerging trends in genome editing. The session on both the days not only opened our doors to new avenues in the big genome world, but also amused us with the fact that there is so much explored still in this world. As quoted by our Honorable Founder President, Sir, Dr. Ashok K. Chauhan, Sir, I breathe innovation, I dream innovation, and ensure that our outcome-based research and innovation ecosystem enable us to achieve ever-rising benchmarks of success that we witnessed in the very first talk of the inaugural session by Dr. W. Silvamurthy, Sir, President, Amity Science Technology and Innovation Foundation, Director General, Amity Directorate of Science and Innovation, who shared with us the glimpses of Amity universe. Followed by the presentation by Dr. Amit Kharakwal, Sir, Deputy Director, Amity Institute of Microbial Technology, who talked about the research and technology transfers done by the faculty members and the enthusiastic students of AIMT. The scientific session 1 was started with our chief guest, Dr. Shubhra Chakrabarti, Director NIPGR, who told about the system-level understanding of organelle control of multi-host resistance in fungal diseases. She told about the role of DAF16 and IRF in generating immune response. Followed by Dr. Ashutosh Pandey, also from NIPGR, who talked about the genome editing tools in food crops for enhancing its nutritional value and securing the food for longer duration. He told about the transcriptional regulations of flavonoid pathway and tools like GCMS, LCMS, which are used to characterize the metabolites. To enhance the scientific discussions and understanding further, the scientific session 2 had 3 fabulous scientists, Dr. Shailesh from NIPGR, Dr. Anil from ICAR, National Institute of Plant Biotechnology, and Dr. Ralph from Germany, who are the experts in genome editing. They opened our concepts about RNA molecules that can produce hybrid genetically modified crops of better quality. Novel molecules like fusion transcripts are the future of plant biotechnology, as told by Dr. Shailesh. Genetic engineering and genome editing for crop improvements, providing the better shelf life, nutritional value, and disease resistance to crops by identifying the genomes of crop in question by finding in it CRISPR, as guided by Dr. Anil. He also spoke about the development of cyanide-free cassava. Dr. Ralph in the end told about the rhizosphere monitoring via self-oligomers that transfer information from root to tips, explained by CTCOC1 mediated dephosphorylation. The session of day 1 ended with the informative poster session by participants that further enhanced the knowledge in the field of genome editing. Day 2 began with the scientific session 3, in which esteemed scientists like Dr. Shashi from ICGAB, Dr. Vibha Tandon from JNU, Dr. Ankush from BHU, and Dr. Manoj from Amity University gave their thought-provoking talks. Dr. Shashi talked about increasing algal biomass by GM technique, which can be utilized in biofuel production. Followed by Dr. Vibha Tandon, who enlightened us about the development of new molecule PBEF effective on multi-antibiotic resistant bacteria specific to bacterial topoisomerase 1A. Followed by Dr. Ankush, who spoke about the alternatives of antibiotics like antimicrobial peptides, probiotics, silver nanoparticles, bacteriophages, etc. He also gave the insights about bread technology for recombination. Last talk of the session was given by Dr. Manoj, who spoke about the effect of rising temperature on plant growth and their functioning, leading to decreased food quality and quantity. He enlightened us about the temperature sensors in plants to develop the climate-resilient crops. The last scientific session included marvelous talks by Dr. Malik, Amity Institute of Neuropsychology and Neurosciences, Dr. Maitri Pradhan from Germany, and Dr. Shivani Sharda from Amity Institute of Biotechnology. Dr. Malik enlightened us about the REM sleep in detail in various animal models. He also explained how we can regulate the gene and control the neurotransmitter in case of REM loss. Followed by Dr. Maitri, who explained that some argonaut proteins are responsible for Nicotiana growth and pathogen resistance, also their involvement in competitive fitness and mycorrhizal colonization. Last talk was delivered by Dr. Shivani Sharda, who spoke about the application of optical modules in optogenetic and optoproteomic genomic modulations. After the 2-day session, it can be hence concluded that the conference was able to meet its objectives and outcomes. It helped us understand the powerful biotechnological tool of genome editing through the intellectual talks, lectures, and vivid discussions. With this, every member and student of Amity Institute of Microbial Technology feel enlightened and aware to pursue and explore this subject furthermore. Thank you.
Thank you, Shalini ma'am. Now it is the time we acknowledge the talent of our students for the amazing and creative posters they had presented in yesterday's session. I request Dr. B.N. Malik sir, Director, Amity Institute of Neuropsychology and Neurosciences, Amity University, Uttar Pradesh, to present the certificates to the position holder. I request you, sir, to please come upon the stage to present the certificates for the winners. From the PhD category, the winner is Divya Chowdhury. Please put your hands together for Divya Chowdhury. Please put your hands together for the position holder. Congratulations, ma'am.
Now, for the graduate and postgraduate category, the third position goes to Ananya Saxena. The second position is secured by Uditi and Ashwarya. And finally, the first position goes to Hafiza and group. Congratulations to all the position holders. And thank you, sir. Gilbert K. Chesterton said, and I quote, "I would maintain that thanks are the highest form of thought and that gratitude is happiness doubled by wonder." With this thought, I would now like to invite Dr. Naveen Chandra Joshi, Assistant Professor, Amity Institute of Microbial Technology, for the formal vote of thanks. Over to you, sir.
Hello. Good evening to all. Thank you is such a prayer that cannot be seen or touched. It must be felt by heart. I, Dr. Naveen Joshi, feel honored and privileged to get the opportunity to propose a vote of thanks on this special occasion of the two-day international conference on emerging trends in genome editing. Let me, first of all, start by giving glory to the Almighty God for making this conference a resounding success. I would like to thank all the honorable delegates and invited speakers who blessed us with their presence by sharing their views and research with us. I would also like to thank all the participants who include invited dignitaries, faculties of various institutions, research scholars, and students. I'm also very much thankful to all the organizing committee members. Words are not enough to thank their constant guidance and support to shape the conference. A big round of applause and my special thanks to Dr. Aarti Mishra for her tireless effort to make this event successful. Please. I am very much thankful to the National Sponsoring Agency, Department of Biotechnology, Government of India. It would have been impossible to organize the conference without the financial support from the DVT. We are immensely grateful to Honorable Founder President Sir, Dr. Ashok K. Chauhan, for his blessings and motivation to carry out such important events. Special thanks goes to Honorable Chancellor, Dr. Atul Chauhan Sir, and Honorable Vice Chancellor, Dr. Balvinder Sukla Ma'am, for their great support and encouragement. Our heartfelt thanks goes to Professor Ajit Barma Sir, Group Deputy Vice Chancellor, Distinguished Scientist, and Professor of Eminence, Amity Institute of Microbial Technology, for his constant support and guidance throughout the preparation of the event. I am very much thankful to all our faculty colleagues and non-teaching staff members who always stand by us and motivate us. I feel proud and thank you for making this event a successful one. An event of this dimension cannot happen overnight. The wheel starts rolling months in advance. We have been fortunate enough to be backed by a team of very motivated and dedicated students and the volunteers. I cannot thank everyone enough for the involvement they have shown and the willingness they have expressed to take on the completion of the task beyond their comfort zones. A big round of applause and a special thanks to our anchoring team, especially Shivani Sarma, Kuldeep Jain, Isika, and Pavitran. Please. Thank you guys for making this program successful. Last but not least, very warm gratitude to the IT department for providing the best platform to deliberate the event. Finally, I leave you with this inspiring quote by Martin Luther King. An individual has not started living until he rises above his individualistic concern to the broader concern of all humanity. Once again, I thank you all for your attention. Thank you very much. Thanks.
Thank you very much, Naveen sir. Now I take this opportunity to thank each one of you for your active participation in this 2 days event. I would request all the esteemed dignitaries, delegates, and all the attendees to proceed for the networking tea. I hope you all enjoyed this program. Now we will be taking a group photo right now. So I request each one of you to come on to this stage for the photo. So this is Pavitran
And Ashika.
signing off.
Thank you.
Thank you.